like to say a special thanks to those of you who are with us from distant time zones. I know, for example, Paula Dawson-Martins and Wits Lendra were up until 2 a.m. in the Netherlands, and actually even did some extra running of genomes through DRAM yesterday for some extra credit. So shout out to them and to all of you who are visiting us from far away. I made a Google Doc that contains a bunch of links, and thanks to Aditi Sengupta for the great suggestion in our feedback channel over on Discord. I will share the link of that Google Doc with you. If you want to check that out, it'll have a lot of links to some of the materials that were presented yesterday, and we'll continue compiling those links as we get through the presentations today and through the rest of the week. And there's also some general links there that give you some information on how to access KBase or how to get to the project web, the summer school website and so forth. So with that, I don't want to take too much of the time away from our presenters. Our first presentation is going to be given by James Stegen and Vanessa Garayburu-Caruso, and they are going to be talking about metabolomics in river corridors. And so James and Vanessa, please take it away.
Thanks a lot, Tim. Also, just a logistical thing, I think questions specifically come through the plenary session channel in Discord. Is that right, Tim?
That's correct, the plenary channel, and then there's also probably a lot of discussion in the metabolomics channel today.
Great. Excellent. Yeah, welcome everybody to the second day of the EMSL Summer School. As Tim was saying, really appreciate everyone joining us. As Tim mentioned, today is about metabolomics in river corridors in particular. And I'll give you just an overview of what we're going to do in this particular talk, and then we'll move from there. So this talk is going to start with some overview thoughts on kind of orienting us to how we think about metabolomics, which is not necessarily in a very traditional way, a little bit different than you might be thinking about metabolomics. I'll describe that in the next couple of slides. I'll give a little bit of context such as that and some motivation elements. And then Vanessa, one of our colleagues here, is going to give you more of a science talk on how we've been looking at metabolomics and linking them to biogeochemical function. And then I'll come back just at the very end to give you an orientation to the rest of the day. There's been a couple of changes in the schedule, and I'll detail those at the end there. So let's see. So with that, and we'll also have a few minutes of Q&A after the end of this presentation. So, okay. So a little context on terminology to start with. So as I mentioned, we're not taking necessarily a very precise traditional approach to metabolomics. We take a very broad view. So traditionally, metabolomics, if you think about that, usually people think about small molecules, central metabolism, and sometimes, you know, metabolomics is split into those metabolites within as well as outside of cells. We take I guess a broader view on this. We're examining small molecules in environmental samples, generally almost always water-soluble organic molecules. And we conceptualize those as metabolites in the environment, and those are coming from diverse sources. They come from microbes, they come from plants, they come from other kinds of organisms. And we really think about them as the organic molecules that fuel as well as are produced by metabolic processes. And Vanessa, if you could go to the next slide, please. I should come through there. Yeah, so and from that perspective then, taking that broader view of metabolomics, and often what we're looking at is our secondary metabolites, but we don't quite parse it in terms of within and within cell versus extracellular metabolites. But generally, as I was saying, looking at water-soluble small molecules in the environment that are biogeochemically and metabolically relevant. And you'll hear a lot today about FTICR mass spectrometry. And this is one tool that we use in particular with respect to Wonders, as people are aware. You know, that's the sort of the data foundation for the summer school. So Wonders has so far focused on FTICR mass spectrometry for doing metabolomics, and that stands for Fourier transform ion cyclotron resonance mass spectrometry. It's a long name. I'm not going to go over the technical details of FTICR at the moment. Will Kew, during the virtual tour today, will give you a lot more technical information on the instrument itself, and Vanessa will go through more details in terms of what the data look like and how we're using them. But I want to really emphasize that this is just one tool in the toolbox. And by no means is it meant to be the only thing that we use. It's a great tool for doing untargeted analyses of complex environmental samples. It's very, very high mass resolution. We can assign molecular formula. We can do a lot of interesting things that you'll hear from Vanessa about once we can do that. And so, but I also want to point out that FTICR is not the silver bullet for metabolomics by any means. It's not quantitative. It does not give you concentrations. And it misses the very small molecules, many of those that would be traditionally associated with metabolomics, and in particular those molecules tied to central metabolism, such as glucose. And ideally, FTICR is paired with other methods, such as NMR, nuclear magnetic resonance, LCMS, or liquid chromatography mass spectrometry, gas chromatography mass spectrometry. Those are other methods that all have their own strengths and weaknesses, and as a collective can provide a broader view of metabolomics in environmental samples. And Will Kew will also touch on those additional technologies, even though we won't be working with those kinds of data today during the summer school. So Vanessa, if we could go on. So that's a little bit of just orientation in terms of terminology and the instrument that we'll be focusing on today. I want to give you some broader conceptual context of why do we focus on, why are we interested in metabolomics from this particular point of view? And there's for a number of reasons, just a moment. So a number of reasons, and one of them has to do with the data when viewed in this way carries a lot of information with respect to microbiology as well as biogeochemistry. And this image here on the left is just an example of linking metabolomics data to microbial ecology. And I'll just briefly go over this, just touch on this, where the vertical axis here has to do with the strength of deterministic ecological selection that is imposed upon microbial communities. So as you go up the axis, selection becomes stronger. And the horizontal axis has to do with the thermodynamic favorability of the metabolites that are in those systems, in those samples. And you can see there's a relatively good relationship between the strength of selection that is driving the ecology of microbes and the thermodynamic properties of the metabolites that they are interacting with, both as resources and products of their metabolism. Vanessa, if you could advance there. And then in addition, the same horizontal axis having to do with the thermodynamic favorability, and this is something that Vanessa will tell you more technical details about, and Hyun Saab will go into even further details of the theory tied to the thermodynamics. And here we have the vertical axis being respiration rates, in this case aerobic respiration rates, being related to the thermodynamics. And so we see this over and over again in our work, where we look at metabolomics through this broad lens, we see it connected very strongly over to the ecology of microbes and emergent biogeochemical function. So, and Vanessa, if you could go to the next slide, please. We can go beyond that, linking metabolomics to other kinds of omics, and this is just a real, just sort of scratching the surface here. On the left-hand side, we have an ordination of mass spectrometry data, the FTICR data, the metabolomics data. Those are the circles, and then those are tied to metagenomic-based inferences of microbial taxonomy. Those are the arrows, and we can see some correspondence. And these are data coming from the WONDERS samples that you all have been working with yesterday, and we'll work with more today. So we can see correspondence between the microbial taxonomy and the metabolomic profiles. And on the right-hand side, we can generate functional hypotheses. And this heat map here, this is the 7 different rivers tied to the WONDERS sampling campaign that was described yesterday. And the colors have to do with the frequency of observations of different EC numbers that we're linking or inferring from the metabolomics data. And so you can start to get a sense of different rivers have different kinds of profiles, and we can generate functional hypotheses about what might be going on metabolically there from the metabolomics data. And that's work from Bob Danczak. You can speak to him in greater detail about that. In addition, we think a lot of our work is tied to understanding fundamentals, but also driving towards enhanced predictive capacity at a broad range of scales. And so another really interesting aspect of looking at metabolomics in this broad view is that we see real coherence in structural organization that in this case, as I'm showing here, is emerging at the continental scale. And we often think about metabolomics as, in small samples, in biological cultures and things like this. But if we view it this way, with broad sampling, this is tied to the summer sampling campaign that Amy Goldman mentioned yesterday very briefly. We can see emergence in structure of thermodynamic properties. Here on the right-hand side, the vertical axis is a thermodynamic parameter lambda that Hyun Sa will tell you much greater detail about what that is. Basically has to do with the efficiency of organic matter turning into microbial biomass. And so if we look at that bounding box on the left, those samples from the Wonders campaign, if we take those data, get the median lambda values from all the metabolomic samples and relate them to longitude, and we see a pretty darn good relationship there, considering that all those samples came from many diverse kinds of river systems with all kinds of different impacts. And so we see structure, and that's really encouraging in terms of being able to use the knowledge we gain from metabolomics to inform models, even working at continental or larger scales. In those 3 slides, I want to emphasize something here that we're showing you really the newest stuff. Okay, I don't know if people noticed, but I had on the lower right there, in prep. Those 3 slides are from papers, manuscripts that are in prep right now. And so we're opening the door here to a very active and dynamic and constantly changing research area. So we're not presenting like the most polished textbook kinds of materials to you today. Really, we're presenting to you the bleeding edge of what we're doing from a research perspective, which I hope you find to be encouraging and exciting to be kind of exposed to and engage with, and in turn advance those research domains. So Vanessa, if you could go to the next slide, I think that is the start of Vanessa's talk. So I'll leave it with Vanessa. I'll come back at the very end with that. Please, Vanessa, take it away.
Hi, everybody. Good morning. So I'm going to be telling you today the story about my newest paper. It's called Carbon Limitation Leads to Thermodynamic Regulation of Aerobic Metabolism. So first off, my name is Vanessa Garayburu-Caruso. Everyone calls me Vanessa GC or VGC, so feel free to do so as well. So these pictures are kind of a little bit about myself. I have a lot of experience doing lab tests and analytical instrumentation and field work, and today I'm just going to be telling you the story about my newest experiment. And so first, let's start with some background. So organic matter processes are key for riverine biogeochemical function. So within the river corridor, areas where groundwater and surface water mix, hypereik zone, contribute significantly to aerobic respiration of the whole river ecosystem, contributing around 90% in some cases. So because of this, it is really important to understand what processes control this organic matter respiration in the hypereik zone. There have been many biogeochemical models trying to integrate what we know so far into local scale reactive models to try to then understand the processes that govern respiration at a global scale. However, we currently have a knowledge gap, and the knowledge gap is driven because a lot of these models consider carbon just like a bulk pool and only represent very simple compounds that do not do justice at the variety and diversity of organic matter compounds that we can find in this hypereik zone. So in order to bridge this gap, we need to have a better understanding of how carbon thermodynamics, concentrations, and the ratios between carbon and nitrogen are affecting this overall respiration rate. And so in order to do this, we have a great tool, which is the FTICR-MS that James mentioned in the introductory slides. So briefly, the way that it works is that you go to the river or you have your lab experiment and you take your sample. Then the sample is sent to EMSL for analysis. And the way that the data looks like, it's like this graph that I have here, where we have on the y-axis is an intensity of the peaks, and on the x-axis we have the mass of the compounds that are identified, or the M/C. These masses are extremely accurate. That's one of the greatest benefits of FTICR. And so because of this accuracy, we are able to determine that this molecule with this mass is precisely the one that we observe over here. So the way that we process the data is that after we have all of these spectra, which match them to a database, and we match these M/Cs to a molecule with an error lower of 1 chemical shift. So with this information, then we can try to calculate thermodynamic properties of the carbon molecule. So one of the ways that we do this is by calculating the nominal oxidation state of carbon. The nominal oxidation state of carbon utilizes the number of atoms in the organic molecule that was identified to calculate just the oxidation state of the carbon. Then using an empirical relationship derived by Larroux and Van Capellen, we can utilize the NOS value associated to that identified molecular compound and then gather a Gibbs free energy. So what we notice is that these Gibbs free energies are positive. And the reason for this is because we need to pair them to an electron acceptor in order to make the overall chemical reaction negative and be favorable from a thermodynamic standpoint. However, in some cases, you have a dominant electron acceptor. In my case, would be oxygen, because we're estimating aerobic relationships. So we can derive a lot of conclusions only on the Gibbs free energy of the carbon by itself, assuming there are constant electron acceptor conditions through the whole reaction. Once we have the Gibbs free energy, we can also estimate something that we call the biochemical transformations. So briefly, we have this compound over here that we showed in the previous graph, and then say we have this other peak that has an identified compound. We have a code that it's calculating the mass difference between each of the peaks identified in a specific FTICR spectra, and then these peaks are mapped to a reference database, and depending on the difference, we can associate this difference to the gain or loss of a specific carbon molecule. So in this case, the difference between this peak and this peak is associated to the gain or loss of a valine molecule. And then depending on how many times these molecule transformation appear, you can also relate how many times this specific molecule could have been gained or lost within a specific sample. So some examples of where we have used these methods are in one of Emily Graham's studies in 2017, she observed by extracting the water-soluble fraction of riverine sediments that thermodynamically favorability was correlated with respiration rates, where thermodynamically favorable compounds were preferentially oxidized. In addition to this, Stegen et al. In a study in 2018 proposed a different conceptual model where carbon thermodynamics and concentration can be driving the respiration during groundwater and surface water mixing. So in his conceptual model, we have our groundwater in our system, which is aerobic, and it has more favorable organic carbon, but at lower concentration. And then we have the surface water in our system that has less favorable organic matter at higher concentration. And so the model suggests that when these two compartments mix, there is a combination between the concentrations and the thermodynamics that we are observing in the system that are driving the higher metabolic activity. So this graph over here is showing the distribution of carbon thermodynamics within our groundwater and surface water, where blue is completely surface water and purple is completely groundwater. Then we also have observed the importance of organic nitrogen cycling in hotspots. So a different study from Emily in 2018 has observed that reactions involving organic nitrogen were higher in areas that present also higher metabolic activity, while they were lower in areas of lower metabolic activity. This was also observed in a Stegen study where we have, I realized that the slide moved around, but where we have an overrepresentation of nitrogen containing transformations in the hypereik zone versus nitrogen-free transformations in the river, being the hypereik zone the area of higher metabolism. So with this, the only thing we still have to understand is in a control lab experiment, how do carbon organic matter thermodynamics, concentration, and carbon nitrogen ratios are influencing aerobic respiration rates. So for this, we conducted lab experiments where we collected sediments from the shallow hypereik zone in the Columbia River, and we took these sediments to the lab. We performed sequential extractions to induce carbon limitations. So this is a graph of the dissolved organic carbon in the extracted water for 50 hours until we induced carbon limitations, and we incubated these sediments. So our incubations consisted of 100 bioreactors where we had 4 treatments, 3 concentrations, and controls. And so in order to understand how the dissolved organic concentration was influencing respiration rates, we selected 3 different range of concentrations. The lower end mimics the concentrations we find in the groundwater in our system, while the higher range is similar to concentrations observed in more urban streams or streams with higher dissolved organic inputs. To understand carbon and nitrogen ratios, we selected model compounds that were either nitrogen-bearing or nitrogen-free. And lastly, to understand carbon thermodynamics, we selected compounds that mimic the extremes observed in our system, again, where blue is the surface water like thermodynamics, and the purple is the groundwater like thermodynamics. So we added all of these compounds into our bioreactors. This is how our bioreactors look like, and we incubated them while shaking for 6 hours at 21°C. We then estimated respiration rates as the dissolved oxygen decrease over time. So we measured dissolved oxygen non-invasively during the course of the incubation. So this is an example of how we did it. We had one dissolved oxygen dot and 1 pH dot, and with these fiber optic systems, we were able to estimate how dissolved oxygen change over time. And then we calculated the respiration rate as the slope in this regression line. So it is important to note, like I was mentioning in one of the previous slides, that our bioreactors stayed aerobic during the whole time. So in here, we can see that the initial dissolved oxygen concentration starts around 9.3 milligrams per liter. And by 6 hours of incubations, we have, excuse me, that the concentration ends in 3.7 milligrams per liter. Because of this aerobic constant environment, we are able to drive more conclusions on the carbon thermodynamic perspective. And so what we first observed is that respiration rates were limited by dissolved organic concentrations. So here we have on the y-axis the respiration rates, which is the slope of the dissolved oxygen decrease over time. And on the x-axis, we have the concentrations of our treatments. We observed that the respiration rates increase from 0.3 to 3, and then plateau from 3 to 9, suggesting that they were limited by dissolved organic concentration. So to dig in deeper into what the carbon character, the organic matter thermodynamics was telling us, we utilized the water, like the slurry water off of the incubation, and we analyze the FDICR data out of that post-incubation water. And so this is just like calling back to the methods um, we mentioned earlier in the talk. With the FDICR data and the molecular formulas identified, we calculate the nominal oxidation state of carbon and the Gibbs free energy of the carbon. And with this, we observed that with carbon limitation, respiration rates corresponded to organic matter thermodynamics. So this is the same graph that I showed you before. So on the y-axis, we have the respiration rates, and we have the carbon limitation zone is incubations of 0.3 and 3. And what we are observing is here on the y-axis, we have respiration rates, and we have on the x-axis, the Gibbs free energy of the carbon. And we see that under these carbon limitation conditions, more thermodynamically favorable carbon also has higher respiration rates. However, without carbon limitation, we did not see any influence of thermodynamic favorability in respiration rates. We see here are the respiration rates. Here's the thermodynamic Gibbs free energy. But without carbon limitations, we did observe that carbon and nitrogen ratios and nutrient availability explain respiration rates. So we observed that nitrogen-bearing incubations had a higher respiration rate than nitrogen-free incubations. So we took this to the next level and analyzed the biochemical transformations of all of our bioreactors. Once again, the biogeochemical transformations represent the potential carbon molecules that were gained or lost in a specific reaction and how many times this could have happened. And so what we observe is that without carbon limitation, the biochemical transformations shift with nutrient availability. So in this graph, we have an ordination plot where we can see the closer the points are together, the more similar those transformations are. And so for carbon limitation scenarios, we see that there is no real difference between the molecules that could have been gained or lost across both of our treatments. But when we overcome the carbon limitation, we see that there is a clear shift in the biochemical transformations that are represented in this reactor. So with all of this information overall, we can propose a new conceptual model. And in this conceptual model, both carbon, organic matter, thermodynamic, and nutrient limitation dually control respiration rates. So in this model, we propose a scenario where as you are initially, when you have low organic matter concentration, thermodynamic limitation controls respiration rates. And as this concentration increases, the thermodynamics become less important and nutrient limitations start controlling these respiration rates observed. And so this is important to include in biogeochemical models because these will help us better predict and under which scenarios these predictions apply when we try to incorporate higher resolution organic matter into biogeochemical models. And so with that, I would like to thank everyone that participated in this study, and also this was funded by Subsurface Biogeochemical Research, and the earlier plots showed by James are funded by Early Career Award. And I would like to open up for questions. On.
Yeah, so we can definitely do, no, that's great. We can stay on the previous slide. So I'll go over this kind of the logistical stuff right just a couple minutes before 9:15. So this is the time that if folks want to ask questions about the science, the concepts, what have you, we can dig in. I'm seeing some stuff in the chat as well as on Discord. Many of the questions there have already been answered, I believe. There was a question in the chat that I don't think has been answered yet, and it was, let's see. So so this is one to me. So how do you calculate the lambda value that I presented in one of those slides, the one where it was linked to longitude. So that's really, I'm going to pass that question to Hyun Saab if he is available to speak at the moment, but I'll just say the short story is that lambda is tied to the stoichiometries of organic molecules as well as the metabolites, if you will. The elemental stoichiometries as well as their masses, and Hyun has developed a lot of theory around how to do that, to go from those stoichiometric ratios into that lambda parameter. And so Hyun, are you available to give just a really brief answer to that? I know you're going to go in much greater detail the rest of the day.
That is one of the topics that in my lecture in the afternoon, but briefly, the lambda is really calculated based on their energy balance between catabolism and anabolism. So we define metabolic reactions by combining catabolic and anabolic reactions such that their energy, for their energy balance. So lambda represents how many times catabolic reaction should run to provide the energy required for the synthesis of 1 carbon mole of biomass. So higher value of lambda means that so many times of catabolic reaction should run to provide energy, therefore thermodynamic efficient compounds. So I'll give more details later.
Thanks, Hyun. And one thing I'll say is that you'll be, there's R code, R scripts that are available to calculate lambda. And so that should be, I believe, Bob, that'll be part of Bob Danczak's tutorial. If not, it'll be part of Hyun's. So those kinds of scripts are available and can be, will be shared. So I'm seeing some other questions. Oh, actually, one thing I wanted to mention.
A bunch of questions over in Discord too. So do you want me to relay those, some of those to you?
Sure. Can I just hit the ones that are in the Zoom for the moment, and then we'll jump to Discord?
Yep, let's
Okay.
do that.
Okay. Let's see. So one thing I'll say also with the values I was presenting on that slide, those are the median values of lambda. And so what that means is that every sample that you have a large number of lambda estimates. So every organic molecule, every metabolite that is identified in the mass spectra and that you can assign a formula to, we can then generate a lambda estimate. And so for every sample, you have a distribution of lambda values. And what I was presenting was the median for every sample. Okay, so some other questions here that I'm seeing. Let's see. How about, someone asked, can you explain the significance of the hypereic zone? Vanessa, do you want to speak to that, please?
Yeah, so the hypereic zone has a different significance depending on the field that you are focused. But the short story is that it's very diverse in microbial communities. It has a lot of processing. It's considered a hotspot within the whole river corridor. And it is very rich in different nutrients. It has different redox ratios, redox conditions. And because of that, it's responsible of most of the processing that happens within a river corridor. And due to this higher processing, it is really important to study, because if you ignore it, you are ignoring the part that is responsible of most of the removal of contaminants, or most of the CO2 production due to aerobic respiration. And yeah, so it's a key ecological area to study for river corridor science.
Thanks, Vanessa. I'm seeing another one here. How do you disentangle thermodynamics versus nutrient limitation when organic carbon serves as the electron donor? I have some thoughts on that, but Vanessa, do you want to speak to that? I think it ties to your experiment.
Yeah, so the way that we were doing it over here was by looking at this relationship. So it was a very strong, nice relationship where we have respiration rates and thermodynamics correlated when there was carbon limitation. And these respiration rates within nitrogen-bearing and nitrogen-free treatments did not significantly change. And so when we overcome this limitation, we see that this relationship doesn't exist which is contrary to what we observed in the previous graphs. And we also observe that these respiration rates are different between the treatments on nitrogen bearing and nitrogen free. So for this particular experiment, this is the way that we are identifying where the dominance of nutrient limitation or thermodynamic limitation. So I'm thinking James really has like a bigger picture of how can we identify these in different systems that are not these precise experiments. So I can let him take on now.
Well, actually, what we're planning to do as we go forward in our research program is to build on Vanessa's experiment and to do additional manipulations using a finer gradient of organic matter concentration or DOC concentration, and evaluate the relative influences of thermodynamics and nutrient limitation across more of a continuum, if you will, to be able to develop hopefully like a functional form of the relationship between the influence of thermodynamics and organic matter concentration, and how that varies under different nitrogen contents of that organic matter. So kind of looking at the interaction between, like Vanessa was doing, but building on that, the interaction between the organic nitrogen and the organic matter concentrations using a more finely resolved experimental manipulations. So that's our particular approach, but I'm sure other people have additional ideas. So I'm seeing some other questions here. I went back to you, Vanessa. What are some potential applications of your research, of the outcome of your research?
So one of the applications that we had in mind actually when we were designing this experiment was for modeling. So we're hoping that by having specific scenarios where you can consider thermodynamic controls versus nutrient controls, you can expand the way that organic matter molecules are being represented in these biogeochemical models. So we are trying to build on the current and existing theory by introducing more substrate-specific types of models, which I think is something Hyun might talk a little bit more in his talk later on. And so we can consider millions and millions of carbon molecules, but by narrowing down the specifics at which we might have to drive these efforts in modeling in terms of thermodynamics or nutrient limitations, we can improve the predicting capacity of these models. And so this would be like one application that we were having in mind as we were designing this experiment, kind of trying to give scenarios that we can input into these models.
Great. I'm seeing some other questions here. How do you classify the compound classes, amino acids, sugars, using the FTICR mass spectrometer? So I'll take a crack at that, and others may want to jump in. But people have tried to do this using, often using a Van Crevelen analysis is what it's called. So you're basically taking the molecular formula and taking the stoichiometry of those elements and plotting them in a particular way. And then, so basically you have a two-dimensional space defined by those stoichiometries. And then there are some sort of general rules of thumb of where different kinds of molecules fall in that two-dimensional space. And that's this Van Crevelen analysis. You can just Google that. You can see some images of that. And people have developed basically different regions of that two-dimensional space that correspond to things like protein-like molecules or lipid-like molecules. And there's a number of other categories. I will say that we generally have moved away from that. Those designations are, I believe, initially developed from the petroleum industry, and it's not clear that they have really great relevance to natural environmental systems. And so we've moved more towards, in terms of classifying things, classifying them based off of their elemental composition. So do they have carbon, hydrogen, oxygen, or do they have carbon, hydrogen, oxygen, and nitrogen? So sort of binning things based off of which elements exist in the molecular formula, as opposed to trying to define them as, protein-like, etc. I mean, there's still a lot of people do that. It's not clear that's so robust with respect to the FTICR in particular. Of course, other instruments can provide much more refined and clear identifications of specific molecules. And so the FTICR gives you the molecular formula, but it does not give you structure. And so that's kind of it's certainly a limitation of this particular methodology. Let's see. I'm getting another question here. Can you further explain the conceptual model? I think this is a question to you, Vanessa. How do we know when the model shifts from thermodynamic to concentration control?
So right now, we don't have a clear idea of how this curve looks like because we only experiment with 3 concentrations. However, the study that James was mentioning is designed to provide a larger range of concentrations over in this axis and more spatial distribution, aiming to understand how this curve looks. Is it like a straight line? Is it an exponential function? So at the moment, we don't really know exactly when the shift occurs, just because we have limited information. However, we believe that this conceptual model will apply to also the basin scale samples that James was describing. And so kind of the idea that we had over here was that for example, headwater streams that had higher inputs of organic matter concentrations, respiration rate will mainly be limited by nutrient availability. And so these were represented over here by the yellow colors. And then as we move down in the basin, or maybe we have arid system or semi-arid systems that don't have as high organic matter inputs or dissolved organic carbon concentrations, we believe that the thermodynamics of the organic matter will be driving the respiration rates there. But the exact shift, it's still yet to come.
Okay, so I'm gonna cover a couple of questions I'm seeing, and then there's one more high-level question for you, Vanessa. So I'm seeing, can you tell us more about the FTICR mass technique? So you'll hear more about that. This is from Discord. You'll hear more about that from a Will Kew in the virtual tour of EMSL. He'll provide you a lot of technical details. Someone is asking about the lambda calculation scripts. Those will be made available for sure. And let's see. So someone asked about what was meant by the term efficient or organic matter. Is it like carbon use efficiency? So lambda, I conceptualize that as something very conceptually similar to carbon use efficiency. How efficient is that organic matter or metabolites in terms of being turned into microbial biomass? So again, Hyun will dig into greater detail about that. So we have 1 more minute. And this question's for you, Vanessa. I don't think I asked this one yet. What is the meaning of delta G in the figure showing the relationship between concentration and thermodynamics? Is thermodynamics only determined by temperature? Can you say a little bit?
So delta G is the Gibbs free energy of the carbon at standard state conditions. And the way that it is determined, at least under this study, is only based on the nominal carbon oxidation state of carbon. Because we are calculating things at standard conditions, these numbers are relative to a 25 degrees C environment and a 1 bar atmosphere. And they just come from this empirical relationship derived by Larroux and Van Capellen. You can adjust these Gibbs free energy values based on the temperature of your study. And we are currently working on that. It goes parallel to some of the lambda development that we are currently working on, where we are adjusting the Gibbs free energy by temperature and pH. But the way that we have utilized it in this study and the previous studies that I mentioned in the introduction is just under standard state conditions. So we're assuming 25 C and 1 bar.
Okay, I think we're out of time. Thanks for the engaging discussions section. And let's see, actually, Vanessa, will you pop to the last slide, please?
Yeah.
That's the, let's see, we're almost there, a bit of lag. Okay, so the next thing up is Bob Danczak is gonna go through the tutorial of the metabolomics analysis pipeline. That'll be for a couple hours. I encourage everyone to follow along with that. This is a very kind of in the weeds, working with the FTICR data. He'll also provide some details on the FTICR instrument. And then it'll go into a virtual tour. I mentioned this a couple of times already with Will Kew. That'll be quite cool, digging into some technical details and kind of under the hood in EMSL. Lunch break. And then Hyun will talk about substrate explicit modeling. This is where Lambda is going to come in, some tutorial there, and then we'll have a hands-on session. Importantly, I want to call out that a change in the schedule here, where we're going to make this hands-on section a bit longer than it was in the agenda. So that hands-on piece will start at 2 o'clock instead of 2:30. So that's a change, so please take note of that. So I think with that, we're ready to hand off to Bob. Bob, are you ready to go?
Yeah, I should be pretty much good to go.
Okay, take it away, please.
Cool. All right, there we go. So I'm hoping my screen is visible.
You're good.
Cool. So there's going to be a little bit, this can be quite different from the previous plenary type talk thing. So this is a hands-on tutorial, and that means I kind of want all 200 or so participants at least giving their hand to try and analyze this data. And of course, when you get more people involved in some kind of tutorial, the odds of something going horribly wrong exponentially increases. So we're going to try to work through that together. You might see me continually look off into the overwhelming nether off to my right-hand side. And I'm basically just looking at the Discord channel. So how I kind of want this to run is that as we go through these hands-on steps, since I can't really directly interact with you guys, if there are any issues, pressing questions, things that come up, please put them into ideally the metabolomics text channel on Discord. So that way I can kind of start addressing them if they come up. Of course, I won't be able to get to every little nitty-gritty problem that arises, but hopefully we can at least start working through things together. So before I start, I'll just give a quick introduction to myself. I'm Bob Danczak. I'm a postdoc at PNNL, and I consider myself a microbial ecologist who likes to dabble into organic matter a little bit here and there. In order to do so, I had to learn quite a bit about FTICR analysis, and also to help the Wondrous campaign really use it in some unique ways. So before we start, I'm going to actually, let me present my screen. So I will be exiting and entering presentation mode. Actually, maybe I'll just leave it like this. Hopefully everyone can see it. Yay, nay, I hope. If there are any immediate problems with visualization, please let me know. So the first thing, as I give an introduction, everyone should take this time to start downloading some things that they might need or will need if they want to kind of participate along. So firstly, I do have a GitHub page for this ICR tutorial, and it does contain actually a lot of files from previous tutorials that I've given, which unfortunately due to GitHub means you have to download everything at one time. So what I'll do is I'll just copy and paste this into the metabolomics channel, so that way everyone has access to that. Next, there is another program that if you want to participate with the first component of this, which is what's a lot of pre-processing that needs to get done, it's called the molecular formula assignment step. You will need another program known as Formularity, as well as a database to work with Formularity. Now, I do want to add a caveat since I myself am on a Mac. This program only works with Windows PCs. So if you have a Mac or Linux-based machine, You're just going to have to unfortunately sit through it for now. Hopefully there will be some more developments regarding molecular formula assignment in the future. So I will also paste these links into the Discord channel, so that way everyone has access to them. So on this page, there are two things that you'll need. This is the Formularity program and documents, and then there's this database file that you need to download. And you might notice that's quite a large file, which is why I'm kind of having everyone jump on this at this exact moment, so that way we can get these programs downloaded when the actual hands-on component wants to start. Also, I do want to add another note if anyone is unfamiliar with GitHub. The way that you will download this, unfortunately, you can't download individual components. All you have to do is navigate to this GitHub repository and click on code, and then there should be a download as zip option. And you shouldn't need to be logged in or have an account or anything in order to do this. There might, yeah. Oh, yeah, and also, sorry, just a note to those folks who are asking about the Lambda R script. It is available underneath this ICR tutorial GitHub repository. It's labeled as carbonefficiency.r. Now I do want to add a note that this particular version of the script was kind of handcrafted for the tutorial, so there might be some bumps along the way. Okay, cool. When looking at it, but if you guys need help, feel free to email me, contact me, whatever you need, and I can help you walk through that stuff. So sweet. So first off, let's go and give an overview of what this tutorial is going to look like. So first, I'm just going to give you a quick glimpse at what the FTICRMS is and what it does, how Wonders uses the FTICRMS, and how we can start accessing the data that Wonders has, provided to the greater community. Then beyond there, we're going to walk through some of the more common data analysis pipelines. So we're going to look at how we can pre-process the data using a R package developed by many folks at EMSL, and it's a super great package, but we're going to use that. We're going to look at how to generate those Van Crevelen plots that James briefly mentioned, so that way we can look at what I like to term the molecular formula landscape across samples and across data sets. And we're going to start comparing some of the molecular properties, so things like the Gibbs free energy or NOSC values that VGC mentioned, across samples and again across data sets. We will then be going into some more higher level analyses. In principle, we won't actually be doing this hands-on, but I do have scripts available for you to use these kinds of techniques. So we'll look at common multivariate analyses like PCAs and NMDSs, the transformation analysis, and I'll briefly introduce the lambda theory that Hyun will discuss in much greater detail this afternoon. I do want to add a note that I forgot to mention. On the GitHub page, when you go to the EMSL Summer School 2020, there's actually both this presentation that I'm currently giving available, and then also this text document, the docx file here. This actually gives a step-by-step walkthrough instruction of what how to run each step. So if you get lost or get hung up on any component, you can refer to that walkthrough kind of to get you back on track. Or if you have to leave for whatever reason, it's a long tutorial, this will also help you get back on track. So cool. So first up, as you heard, FTICRMS stands for Fourier Transform Ion Cyclotron Resonance Mass Spectrometry, which is exactly why we abbreviated FTICRMS, because that is a mouthful. And it is capable of detecting many different carbon compounds due to its incredibly high mass resolution and accuracy. There is a link within the PowerPoint that takes you to a low level, but great overview of how FTICRMS how the instrument actually collects data. And you can access that at the Mag Lab. In principle, all it does is measure it ionizes some sample using many different ionization techniques. Those ions enter into a cell. We can then measure the mass of these ions, and then using these masses that we get out of it, we can assign molecular formulas using previously compiled databases. Today, we're not going to be focusing on how to take data that's directly from the instrument and actually processing it. We're going to have data that's already undergone one level of pre-processing. We're going to cover how to download some of this data from existing sources, how to get it into a more usable format, and then how we can start generating figures using this data. The reason we aren't covering the actual machine to processed data pipeline is because there's a lot of considerations that need to be made when doing that initial pre-processing step. And it requires a little bit of nuance that we don't unfortunately have enough time, or unfortunately due to some resource restrictions, the resources to actually carry out rather. So how does WONDERS really kind of mesh with this FTI CRMS data? Well, WONDERS, as you've heard yesterday and I think earlier today, stands for the Worldwide Hydrobiogeochemistry Observation Network for Dynamic River Systems. Again, WONDERS. And we collect samples from many hydrologically dynamic rivers throughout the world using distributed sampling and very close relationships with many, many collaborators. So as part of WONDERS, we collect copious amounts of different data, but one of the key things that we do collect is FTI, well, I don't want to say one of the key things, that's unfair to the other data types. One of the things that I'm interested in is FTI CRMS data that we collect for each sample. This data, once we have it run through that initial pre-processing step that requires some consideration and proprietary software, we upload to a public repository, ESS Dive, for public use. As part of WONDERS, we do take the onus to make sure that that data is QA'd and QC'd, and that the data makes as much sense as we can, so that way all the any researcher across the world can take this data, analyze it, and answer questions that interest them. Now, I do want to stress when I say the data makes sense, it's just to make sure there isn't any outstanding outlier or problems due to data collection. We don't really do any kind of like analysis to ensure we're steering analysis in a specific way. So the first good starting point, so that way anyone who's watching right now can actually kind of jump in and start analyzing any data that they may have seen that's really interesting to them, is how do we actually access data from ESS Dive? So it is all available on the data archive, on the DOE's data archive, which is called ESS Dive, which I will also post into, oops, sorry. Apparently he decided to copy as an image. That doesn't make any sense. Copy. Unfortunately, I will have to do a shortcut because I already
I'll put it, Bob, I'll put it in there.
All right, yeah. Oh, okay. I already actually had it open in another place too. Pre-planned. Yeah, so to walk through it in a PowerPoint format, and then I'll actually walk through it in the how I would find it, because I can never remember the ESS Dive format. First thing that you need to do is navigate to this actually ESS Dive repository, and then when you see it under the search phrase, simply type WONDERS. When you type WONDERS in there, you will see many different entries appear, and each of the entries that you do encounter are things that the WONDERS team has uploaded. And when you click on it, you will see something that looks something like this, with the authors, the name of the data set, a DOI, so it's actually very citable, and we encourage you to actually cite it if you do use this data. But more importantly, you do see the two metadata file, and then the actual data package, we call it. And this contains all of the FTICR data, geochemical data, and anything in between, hydrobiological data. On this page, you'll see a download link. In order to actually download data, you do have to have an ORC ID, ORCID, however it's actually pronounced. But when you do have that and you are logged in, you can then simply just click download, and it will download. You can either click, click download here or download up here to either download all the files or just the data package. When you download it, it will look something like this, where we have WONDERS 48-hour sample site. Well, I should say WONDERS if it is the 48-hour sampling campaign, 48-hour, and then the sampling site. FTICR, geochemistry, metadata, MPOC, provisional hydrograph, or evening site photos. Naturally, since we are in a FTICR focused tutorial, the key data of interest is contained within the FTICR folder. And when you click on that folder, you will see a number of different files in there. Before I move on, actually, I want to show how it actually works. You'll see a number of different files in there that are labeled XML. So to kind of show this manually, how I always have to find ESS Dive, because I can never remember the website, is if you simply just type in ESS Dive to Google, go to it, you'll see a data portal up in the right-hand corner. When you click on that data portal, it will take you to the ESS Dive website, data archive website, I should say. From there, under search phrase, all you need to do is type in wonders, enter, and then it will pop up with all of the data sets that we have collected. And I think this already should have filtered it to the wonders data that we have. Yeah, it has. So anywhere on the map is things that we can see we have wonders data that are provided. But then we can also just click on the wonders data itself to take us to one of the rivers. In this case, it's the Altamaha River in Georgia. You can see it has how many citations it has, so you can find out what papers it was cited in, how many times it's been downloaded, how many times it's been viewed. You can either, in this case, for some reason, download all is not highlighted, I'm not sure. From there, you can see the download button. Wait, where is actually? Something must have went. Oh, it's because I'm the author. Let's not worry about that one right now. That's why things looked a little bit different. Yeah, so let's go to this one just for demonstration purposes. Then you can see something here. We can actually click and download it. So regarding Britt Abrahamson's question in Discord, do you need to download the ESS Dive data for the tutorial? No. So I actually do have demonstration data already provided for us within the GitHub repository, because it's actually data that you will be looking at later in the day. So I took the liberty to kind of parse some of that data out for you guys already. Kind of just showing you how you would actually download some of this. So once you're here, then you would just click on download. Don't want to download it at the moment because that's a bit of a big file. But once you get that zip file, you will be able to see all of those different folders and file structures. And you're free to use those in whatever, with correct citations, in whatever way you find most interesting. So are there any questions about actually ESS Dive at this moment before we kind of jump into the first hands-on component? If not, I'll just kind of roll along.
I'm not seeing anything in Discord or the chat as far as I can tell. So I think you're okay for now.
Sweet, 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 sweet. Is there a way to bulk download? Are you referring to download from all of the Wonders data? So like every single thing that Wonders has ever processed? I don't believe there's a way to download everything immediately, but you can. There's, I think, 9 data sets that are currently uploaded. Fortunately, that's not too hard. But no, there's not a way that I'm aware of to actually download all of these data sets at the moment. There might be a API or something in the back end that you can actually access. I'm just not aware of it. Yeah. Can I still follow the tutorial without the formularity part? So yes, Kadir, you will be able to follow the tutorial immediately following the formularity part, because as part of the GitHub repository, actually I'll kind of take you there to orient you to that. So within the GitHub repository, there is a folder called XML files, which will be necessary for the formularity part, but I have taken, again, the liberty to pre-generate that report that will be created from those XML files for anyone who is on Linux or Mac OS to kind of follow along once that formularity component is done. So the key reason that we do want to kind of give some tutorialization information about how to use formularity is because when you download the data from ESS Dive, it is going to be in this XML file format. And we want to make sure that folks have some ability to at least, even if they can't do it today, they understand how to analyze it in the future. I do want to add another note that there are currently other ways in development to do this step. They're just not currently ready. And there is, I don't want to like tip a hat or anything, but there's ruminations of trying to implement this type of step in things like K-base to make it more broadly accessible, to make it more usable. Now again, there's no official movement on that regard, but there are, we're trying to make this step as amenable as possible, as broadly accessible as possible, I should say. So cool. So as I mentioned, when you download stuff from ESS Dive, yeah, it's a big file. That's kind of why I was hoping to stall for as long as possible. Yeah, so when you download the data, either from the GitHub repository or from an ESS Dive data package, it will be in these XML files. So these XML files are what we call peak picked. So as VGC mentioned in her talk, the data that comes off the instrument after there's actually some initial pre-processing that goes into it before that, exists as a mass spectra with intensities on the y-axis and m/z values on the x-axis. We could go through and manually pick what we think are good peaks, but that becomes problematic when you have thousands upon thousands of peaks to go through in hundreds upon hundreds of different data points. So there are proprietary programs that exist that will use an algorithm to kind of identify the noise, basically identify what's a real peak from the background noise. So we do take that step, we perform that step first, just to make sure that folks don't need to have access to some proprietary software or do something that's a little bit unusual. So before I answer a question, I do want to just add what formularity is actually doing, and then I can kind of pause for a second. So for the data that is in ESS Dive to actually be useful, it does need some initial processing using a program called Formularity, which was designed by an excellent researcher at EMSL known as Nikola Tolik, as well as many other folks. But this program is quite versatile, and it will allow us to calibrate peaks, assign molecular formula, align data, and allow us to analyze all of this data kind of together. So what do I mean by calibrate and alignment and things like that? So the data that comes off the instrument, while pretty reliable, we kind of need to give it, at least under the current envisioning of FTICR data analysis, we need to give it a point of view. So we need to make sure that all the data that we're analyzing together is analyzed under this common framework, basically. We need to ensure that similar peaks are identified within each of these data sets, and that we adjust the masses to ensure that they're directly comparable to each other. Alignment is then a step which is questionable. It's necessary at the moment, but it's a complicated step. We basically take a moving average of all of the masses and start combining combining entries, detected peaks, below some common threshold. There's nuance to it, but that's ostensibly what it is doing. Using these then finally derived masses, we can then assign molecular formulas according to that database file that you are downloading. So this is where I do want to add an important caveat about FTMS derived data. This data only provides molecular formula. It does not provide compounds, does not provide, we will use the term metabolite, but even then it technically doesn't provide a metabolite, as we have no structural information about these molecular formula at all. We can infer structural information using the, some properties of the formula, but it is still inherently a molecular formula. And so the language that you use around it needs to be tempered as such. Another important caveat, as was alluded to before, is that this data is not quantitative. So when you look and first analyze this data using formularity, you will see many intensities for each of the different molecular formula. And it might be very tempting to use these intensities as a sort of abundance measurement. There might be some very limited cases where that's fair, but I would say for the general, especially for environmental data and for most general analyses, it is prudent to convert those intensities to simply presence-absence. And so those are the two main caveats I want to add with FTICRMS data when using it. There are downsides, but there are many commiserate upsides to FTICRMS data that, so trade-offs basically exist. Ostensibly, what formularity will do is then from these XML files generate a report that's called, and this report will contain all of the molecular formulas, presence-absence data, as well as some other miscellaneous information. So if you wanted quantitative information, what types of measurements would you have to do? Well, the question is, what are you trying to measure? That really depends on your research question. So one of the benefits with FTICRMS data is it's incredibly untargeted. So we can get access to many molecular formula, whereas there are many quantitative measurements that have trade-offs. You have to have some question in mind, something to target basically that you need to investigate. And as James is mentioning, Will Kew will go into detail, some detail, about what other instruments can be paired with FTICR to get a more complete DOM picture. Because Linux cannot use formularity, is it in KBase or other FTMS analysis? So unfortunately, at this current moment, I don't want to stress, this might change shortly, and I don't want to, again, give any firm numbers on anything. Unfortunately, at this current moment, we cannot do this step when it's not on a Windows PC. There are other programs that do exist that do similar things to formularity currently that do exist. I think there's one that is in Python. But since this is giving the pipeline that Wonders uses, the standardized pipeline that we've used throughout the Wonders process. And we don't want to kind of introduce some more variables to that process as we can go by. There is, as I mentioned, I don't want to say firmly because there's nothing firm currently, but there are efforts to introduce formularity into KBase and trying to get this entire pipeline in a more streamlined fashion that you don't need to kind of have as many different programs. We aren't there yet, but we are working toward that. I know that doesn't directly answer the question, but unfortunately, if you're on Linux or Mac OS, and again, I am on Mac, so I always have to have some access to a Windows PC when I need to do this analysis. I unfortunately can't participate in this first step. Now, I want to mention there are many other steps that you all will have the full capability of participating in regardless of your operating system. Will be interesting because, no, it should work on Linux. I did actually have someone test it on Linux. So yeah, every step should work regardless of operating system, as long as you have R installed. Hopefully that was enough stalling to get at least most folks with formularity downloaded, maybe a database. Hopefully. It's a big file. Yeah, I know, it's unfortunate. let's see. Well, what I can do is kind of give an overview on, yes, and James is mentioning within the Discord chat that there is interest in trying to broaden accessibility to things like formularity. And so we're hopefully getting there. So how often is this database updated? Will we need to constantly reinstall it, Bram asks. As you can see from the number on that database, it should actually still be on that actual file. I want to make sure I'm not speaking. Yeah, so that was updated in 2016. Yeah, I don't expect it to be updated anytime soon. So this is a database that was compiled by folks at the Woods Hole Institute, Oceanographic Institute, a while ago, and it's kind of like the de facto formula assignment database at the moment. Now I want to stress there are, hot off the presses, there are currently new pipelines that don't require this database in development. And I can't speak any more to that, but there are new ways to start assigning formulas currently in development that eliminate a lot of the restrictions caused by this particular pipeline. Minimize that. So what I'll do, twofold. I'll do the same thing that I did with ESS Dive. I'll walk through what formularity is and how kind of orient you to how to use the formularity program. And then I'll actually give a more me running formularity. So that way we can see all aspects of it. And more importantly, to hopefully delay long enough for folks to actually get formularity installed. I do want to add an important thing that I didn't actually directly mention. If you do use formularity for anything, especially if you are taking Wonders data and downloading it, do be sure that you are citing Nicola's paper, just to make sure that credit is given to where it needs to go. So yeah, so as I said like 800 times, unfortunately, formularity does require Windows at this juncture. So those running Mac OS, Linux cannot participate at this moment. The rest of the tutorial, fair game. This exact moment, unfortunately, I'm sorry. So the first two things that the reason formularity is so useful is it's actually a very user-friendly program. So when you first open up formularity, you will get a window that looks a little bit more blank than this. But it's very drag and drop based. So you don't have to do too much like finicky with like command line or anything like that. It's a very well-designed GUI. So the first thing that you want to do is under the, under the GitHub repository, the EMSL Summer School 2020 folder on the GitHub repository, you will see a file that's named neg calibrants pip nt with hmc .ref. This .ref file is the calibration list that formularity uses to kind of start putting all of these different samples onto the same context. Sorry, double checking some things. I saw that there's some action in the other channels and I want to make sure there weren't questions. So yeah, and if you, I don't know if it will work. Yeah, so if you actually open up the calibration list, it's pretty neat. It has some information of where it came from, but more importantly, it's actually a very simple thing where you see a molecular formula and then it's given mass and whatever Z value. And that's ostensibly all that we're doing is we're trying to find masses that match this specific molecular formula in our data set to put all of these data in the same framework. Again, framework in air quotes, it's a little bit more complex than that, but from there, all you have to do is drag and drop that calibration list under the location that says drop calibration file. Pretty nice. Next, then this step will take a moment. That Woods Hole molecular formula database that you downloaded, hopefully, or might not be, maybe downloaded. There's another place that says drop database file. Simply drag and drop that molecular formula database into that drop database file location, and it will load. important note, there are a lot of steps within Formularity that when you drag and drop something into it, might seem like it's just not doing anything. And it seems like your computer is completely frozen. It's not. Trust me, it's going, it's working. It will work. That's an interesting error. I've never run into that error. So Alejandra is mentioning that they cannot drop anything into Formularity. I'm not sure. We will.
I might have a suggestion.
Maybe.
Usually the drop portion gets habilitated after all of the other conditions get fixed. So making sure you have all the check buttons, all the other parameters fixed with what Bob is showing on the screen, and then you should be able to drop. At least that's what has happened to me.
Okay, that's weird. Normally, so we will be going over exactly what you need to check and not check. Usually I was able to drop those first. Oh, well, it doesn't matter. Maybe it's because I've used it so long that my stuff is already set up to be default. So we will make sure that works. There is a step where I will go through what you need to check. Huh. That's interesting. I didn't know you had to have anything checked for that. That's actually surprising to me. Yeah, that's why we have folks that are helping out. So yeah, when you have those dragged and dropped, again, the molecular formula database will take a moment to run, and it will seem like nothing's happening, but it is. From there are a couple settings that you do need to tweak, and I do have these listed out within the walkthrough. Oh, okay, so you do need to change to linear first. Okay, that's a good note. So I guess it comes default as none, maybe. I thought it was defaulted as auto. Under this regression drop-down menu here, you need to change that to linear first. So there are other regression methods that you can use. We use linear due to convention. So the first thing you need to do, apparently, again, this is for a very long time. Am I still on? Okay, good. Sorry, my VPN freaked out for a moment. Yeah, so you just need to take this drop-down menu and switch it to linear. That's a very good point. Thank you. That's pretty awesome. So from there, once you have those files loaded, so you won't be able to do, I know that you won't be able to do this step until the database is actually loaded. You want to make sure that CIA up in the top is checked. This stands for the Compound Identification Algorithm. This is actually the currently the standardized algorithm that follows a couple important rules in identifying whether or not a mass matches a mass within the database. Under calibration, so this box here, there's a calibration section. You want to make sure the start tolerance is set to 5, regression is linear, and end tolerance is set to 0.5. These should, and now again, apparently I might be wrong, but because I've used it for too long that I don't remember what it was like the first time I used it. These should be the defaults, but just make sure if they aren't, make sure they are set to that. Next, under this CIA formula finding step, you want to make sure that here there is a checkbox that says alignment. Okay, good. So yes, some confirmation that 5 and 0.5 are the defaults. Good. Next, under alignment, you want to make sure that this is checked. And then under alignment tolerance, we set this to 0.5. So this is the step that is merging different peaks. Yeah, so you won't be able to check CIA until the database is loaded. You have to ensure that the database is first loaded, or else CIA will not be available to be checked. Because as part of CIA, the actual identification algorithm, you do need to have a database for it to reference back to. So hopefully that fixed it. But again, it will take a second for that database to load. Takes a hot second. From there, there is one more thing that you should need to change. Everything else, the formula tolerance should be 0.5, should be default. dB mass limit is 500, should be default. Use relationship should be checked. If use relationship is not checked, make sure it is checked. And then under the max relationship gaps, make sure that's set to 2. When you have downloaded Formularity, so it should have downloaded an entire folder, there is actually a document that comes along with it. You can open that document to see what each of these tweaks that we're doing really changes. Each of these changes one specific behavior of our compound identification algorithm or other components of Formularity, and they can have wildly profound impacts on the formulas that you get at the end. Something as simple as having the start tolerance set to 6 instead of 5 can change the outputs that you get at the end. So it is important that if you're trying to analyze things in a very standardized manner, that you keep everything consistent across all your data sets or within wonders or things like that. And more importantly, when you do publish, try to be as complete as possible to ensure that people can replicate exactly what you're doing. The last step, if it's not, and I will answer your question 1 second, the last thing on this step that you want to make sure you do under everything else that we just discussed, there's a box that says you use formula filters. Again, pretty sure that's checked by default, but do make sure it is checked. And then under user-defined filter, you do have to put in a whole mess of nonsense there. So this is kind of controlling the limitations on what molecular formula can and cannot be assigned. And again, this is due to some underlying limitations of FTICR that we have to take into consideration when assigning molecular formula, at least currently. Yes, so what I was about to say, since this is a mess of stuff, under the Word document, all of these things are mentioned. So when you downloaded the GitHub repository, why do I keep going there? Have it, that's why. So under the GitHub repository, where it has something like this, but on your computer, the calibration list, is under this one right here. So neg calibrants PIP NT with HMC.ref. So that is your calibration file. The list of settings can be found either by looking at the image under the PowerPoint. So that image is in that PowerPoint file. Or you can open up the Word document. Which looks like that. Oh, and there is one step I forgot to mention. Oop, okay, I'll mention that though. So this will list all the settings that should apparently not be default, but more things were not default than I thought they were, but such is life. But yeah, this walkthrough will list the settings, particularly the one that's the most annoying is the user-defined filter, which you can then copy and paste over. Regarding not checking the CIA, that is interesting. So just to make sure, you do have the Woods Hole database. Well, in your case, it would just be the db.bin file. It doesn't have the GUI part on it. You do have that loaded. And you do have everything else down here as well as use relationships checked. And then also, okay, settings that might not be default, just make sure they are default. Make sure charge is set to 1, adduct is left blank, and then ionization is set to proton detachment. I don't want to go into details about exactly what those mean because they are fine. There are some fine details, but the document that does come with Formularity does outline what each of these behaviors do. So for instance, the ionization step, you would need to change depending on how you collected your data on the instrument. Since all of this data is coming directly from Wonders, we collected all the data in a very standardized way, so we know this upper part doesn't need to change. So loading the database, currently, just due to ease, the database is accessible off of the Formularity website that I provided briefly in the beginning. Yes. As Will Kew said, there is a database included in one of Formularity's public releases. It's a bit of a big file. But all you have to do is drag and drop that database into, oh no. Oh well, I can get back to that. Drag and drop that database back into the gray box that says load database file here. As well as saying, it can take up to 30 seconds, it can take 30 seconds up to several minutes to import, and it will seem like nothing is happening. I want to stress, it will seem like Formularity is completely frozen, nothing's happening, and the folder from which you dragged the database file also will be fairly inactive. I can say with like full confidence, something is happening. it just doesn't seem that way. But so with this in mind, there's one other step. Before I promise, I will then actually walk through how I will actually show you me doing it. So that way we can actually see together all the steps that I do, and then you can kind of align. Because this is right now more of a theoretical overview of how Formularity works. Yes, the box for adduct should be empty, and the result box should be M-P. Yes. So once you have all of that stuff set up, you should have a nice alluring drop spectra files green box in the upper right-hand corner. From there, you will be able to take those XML files that are on the GitHub repository, which of course I closed with Command-Shift-T, Control-Shift-T. Of course, nothing's working. Okay. Okay, keyboard's dead. Fine by me. Yes, so under the EMSL Summer School 2020, the XML files, you can then drag and drop those to that nice alluring green box, tantalizing green box. Again, it will seem like nothing is happening. There is an eerie calm amongst the data. But that's just because it does take a second for Formularity to really load that stuff into memory and actually run through the formula assignment steps. Now, is my keyboard? That's really irritating. 50-50, this keyboard just sometimes shuts off. Okay, so now what I will do is actually walk through how I run Formularity very quickly. Hopefully getting everyone caught up to the last step here. So hopefully folks can see the screen when I'm doing this. So as I said, I do always have to have access to a Windows PC, which means I always remote into a Windows PC. Ooh, that's unexpected. So I'm just gonna minimize this stuff, don't worry about it. Not closing anything. So open Formularity, and bam. So you can see mine's already kind of set up default. But that's because the nice thing about Formularity is once you set these settings once, every time you open up Formularity, they will be maintained default. You'll notice that I've been playing around with another setting here, so I'll just change that back to 5. I cannot see Discord. Can folks see the Windows screen? Preferably James or someone, speak up if you can see your PGC.
Yep, I can see it.
Cool. So, okay, as I said, you can see it looks pretty, mine's already set up to where the start tolerance is 5, end tolerance is 0.5, charge is 1, adducts blank, results M-P, ionization is proton detachment, max relationship caps is 2, use relationships, all that wonderful stuff we covered, it's already checked, and then I do have the user-defined filter already in place. And again, I want to add the significant point that tweaking any component of this can have quite profound impacts on the eventual report. This is what I would consider the standard Wonders settings for any data you collect from Wonders. You need to make sure it looks like this. There is something here that does, I didn't cover under peak filters, the minimum S/N, which I have to 7, but I think defaults to 3 or something. This you don't technically need to worry about as much because the data that we've already filtered that we've published is already filtered to a signal noise of 7. But if you want to feel better, you can change that to 7. So from there, just quickly reorienting myself.
Bob, you have a question in the chat. Should they just drop one of the XML files onto the drop spectra?
You can drop at least 2. If you drop 1, so formularity behaves quite different if you use only 1 file at a time than if you do multiple files at a time. So I would at least do 2 as just demonstration purposes to make sure everything is running correctly. So just to show you what I have a common location for my stuff. So I drag and drop my calibration file, loaded. You can see it says neg calibrants, and then drag and drop the database file to where it says drop DB files. And then as I mentioned, for anyone who might still be loading it right now and might be having some trouble with the CIA, can't click it or anything, you can see formularity is doing a whole lot of nothing. This folder doing a whole lot of nothing. Just kind of got to patiently wait until formularity finishes loading.
Bob, are you gonna tell us again where to get the user-defined filter?
Yes, so you can find that under, can you still see my screen?
Yep.
Cool. I will copy and paste it into Discord. That'll be easier. Copy. Paste. What is going on here? I'm being punked. Every time I copy something from a Windows product, a Microsoft product, it seems to want to consider it an image. So there must be some weird text processing on Discord that I don't understand. There you go. That should be the user-defined filter. But it is also under the walkthrough document that would have been downloaded with the GitHub repository. And so if we go back to my computer, yeah. So you can see the only way the CIA will be able to be checked is if you see the terms loaded formularity db.bin. If you do not see loaded formularity db.bin, you're not going to be able to check the CIA box because you do need to have a database file loaded. Also, you probably, no, okay, never mind. That was me testing. So once you do have that database file loaded, all you have to do is drag and drop it over into the load DB files box. Once you have that loaded, click CIA. You'll see a nice little alluring green box up here. Okay.
Bob, I think I made a mistake because I dropped the DB file into the IPA formula finding location where it said to drop database, but I was supposed to just drop it in the main window.
Oh.
Files. Is that right?
That won't cause a problem or anything. So IPA is another way of assigning formulas. It's a little bit more advanced. It requires a little bit more nuance in order to assign formulas that way. As long as you simply drag and drop it under the CIA formula finding step, and then make sure CIA is checked and not IPA, you shouldn't have any problems.
Okay.
Now, again, I've never done that, but the formulary is designed to have those kind of segmented from each other.
All right. So I was having the same problem with CIA not checkable, and then when I loaded, just now I dropped it into that CIA formula finding, and now it's available. So if anybody else is having that problem, they may have done the same mistake I did.
That's a good point. Yeah. So under the CIA formula finding, you need to make sure the database is loaded. Not under any other formula finding, but CIA. That is the current, as I said, defaults. Not the best, just the default assignment protocol. Yes.
Bob, you have another question about the calibration reference. Is it one calibration reference file for any sample, or does it depend on the targets?
Ooh, yeah. So that's a very good question. So currently, Wonders uses a default calibration reference that was provided by Nikola. It's a broad target calibration list. But if you want to improve calibrations and make sure they are geared toward your system, it would be prudent to design your own calibration lists. Now, it's not like super easy to make sure you have a good calibration list, but it would be prudent. That is something that currently within Wonders, what we've noticed is that there is some difference in calibration and how good calibrations are purely based upon whether or not samples were derived from water or sediment. And again, that's just the only variation between them. It's the same site, but if we have water versus sediment, calibrations change their capability. Now, second note, there's a lot of difference between water and sediment. That could also be messing with the calibrations as well. Complex mixtures are complex, and when you go to sediment or even soil or things get even more complex than water. But it might be, in our case, we are trying to develop a new calibration list that might be able to somehow control for the differences that exist between the sediment and surface water. Might not improve results at all, but at least the calibrations are better. So yes, there is a question of targets, but the one that I've provided is a fairly broad range general negative calibration list for samples collected using ASI negative ionization. Cool. Now I'm just trying to find where I put this data. Cool, and I found it. So yeah, once you have everything set up and there's a nice green drop spectra box here and CIA is checked, if IPA is checked, I quite frankly don't know how it's going to behave. I'm just going to do 5 or 6 or so. Simply highlight the XML files that you want to analyze, drag them over to the drop spectra files, and let go. It will turn red, and again, it will seem like nothing's happening other than that red box, the green box becoming red. But if you wait patiently, you will see that it will change from drop spectra files eventually to a file name. Unfortunately, we'll just list the first file name. But more importantly, within the folder that the folder that contains the XML files, you will now have 2 new files known as report and then report date time log. So the report contains all of the molecular formula information, the intensity information, the combined samples, and then the log actually contains the calibration information. Today, that's not super important, but I do want to add the note. Anytime you download Wonders data, we do have scripts that take into account whether or not samples calibrate good or not. And so you should run those. We do provide those scripts and things like that, so they're readily available to you. But you should download those scripts. And if you are analyzing Wonders data, remove badly calibrating samples to ensure that things don't go wrong. And it's because whenever a sample calibrates badly, what basically happens is its mass is always offset from all the other masses. It's basically, again, the whole point of calibration is to ensure everything is under the same framework. Again, air quotes framework. And if you have samples that aren't in that same framework, they're going to appear wildly different. And they do. Under multivariate analyses or things like that, they always appear almost 100% different from samples that do calibrate well. So we do have ways of identifying them and controlling for them, though. Hopefully, folks, I'm going to move back to my screen here. Yes. Will is adding very good, important information here. So is there any consensus on to whether or not folks have had any luck running Formularity? Because we're into an hour, and I would actually like to move on to the next step at some point to make sure folks can at least understand how to use this information. So if you have a report log, but no, if you have a log but no report CSV, that is Formularity's way of kind of telling you something went wrong. Unfortunately, it might be hard to find out what wrong. Chances are it was a calibration error, which shouldn't happen with this data. I might be mistaken. I'm going to double check. Maybe that's why I had my tolerances set higher. It's possible. So while that's running. Yeah, that's usually an error that happens when there is no things. Because usually the only sample that ever has any problems that we've ever added a deviation from our sample analysis is, yeah, look at that. Yeah, so, okay. We can troubleshoot this live. So as I mentioned with bad calibrations, if we open up that log, don't worry about this. This is stuff that as part of Wonders we do actually take care of for you. It just got, oh, darn it, I'm on a Windows PC. Okay, so all I was doing is, so in order for things to calibrate well, you actually have to have a number of calibration points. And this data that we actually selected as a demo data is the best. It was just for demo purposes anyway. But what you can find is something like this peak here that has, sorry, this, not peak, this sample, which has no calibration peaks associated with it. So what Formularity basically did was it said, hey, there's no data associated with this, this sample. And said, hey, yeah, I'm done. Since there's no data associated with the sample, I can't do anything, so I'm done. So the correct thing to do would be to remove this sample and analyze it, reanalyze it again. Now again, I'm going to stress, this is atypical. We normally do take care of something like that. So what sample was that? Oh my God, Windows. I'm using a Windows keyboard, but on a Mac. And so then when I switch back to Windows, I'm still in Mac mode, and it's all a mess. So Columbia surface water 662. And I'm just gonna move that because I don't want to actually delete it. But if we then rerun it, it should actually run through. But first, this is extra stuff. Don't worry about it. We're just getting it back to the point where I can show. All right. So a good way you can tell if Formularity went wrong or right is Formularity should take a second to run. And by a second, I mean quite a few seconds. If it finishes almost immediately, that means it ran into an error. And so the log will then contain information that allows you to troubleshoot that error. Stress. If you are using Wonders data, those errors should be taken care of. If it's published Wonders data, those errors will have been taken care of. These were data that we kind of threw together for this exact purpose today. Yeah, so that's the problem, is that one of the samples was not calibrating correctly. What I recommend doing, just to make sure everything works correctly, is simply select the Altamaha River samples. So the 6 samples that begin with Altamaha, and then drag and drop those onto the box instead of using all of the data. That would be the simplest approach, rather than trying to troubleshoot it like I just did. But know that the log does contain information, whether you can or cannot troubleshoot it. Yeah, so I'm gonna guess when you only used one XML, as I said, formularity behaves differently, whether you provide it one XML or not, or multiple. What I would take a guess at is that since we have it set to align, formularity probably is like, hey, I can't align a single XML file. Don't know what you want me to do with this. Alignment only makes sense if you have multiple data sets. Now again, so I do want to add the stress. If you're having problems and you're only generating a .txt or a .log file, try using a subset of the data, primarily the Altamaha data. And that will help you climb over that bump a little bit. Cool. So that means everything should be set. Now again, this is where things get a little weird. I can't guarantee that everything is correct, but that's interesting. How are folks getting two different results? That's curious. Let's see. So
Bob, you might think about showing your formularity window and making sure all the settings are correct for the folks getting into their stuff.
Yeah, so here I have formularity open. Hopefully it's clear. If the settings are askance from this, it could throw problems. So one of the key things is do make sure the adduct is blank and that the result is M-P. So that means mass minus a proton. Make sure that you have the calibration set to linear and that the start tolerance is 5, not 0.5, but 5, and that the end tolerance is 0.5. I'm trying to think of things that would be the most likely to cause like a no-run error. Alignment, that wouldn't stop it from running. You would still get reports, but you would get single reports. Formula tolerance wouldn't stop it from running. dB mass limit. Make sure the, one interesting thing that I've noticed sometimes happens, make sure that the minimum relative abundance is 0, and make sure the maximum relative abundance is 1. I've noticed that sometimes this, on some folks, when I've been helping them out with formularity, that randomly doesn't always set to 1, and I don't really know why.
So Bob, I'm wondering, seeing something in the Discord, the ionization must be proton detachment.
Yes.
Right? As opposed to proton attachment.
Yeah, so this has to be detachment. If it is attachment, you're not gonna get nothing. It's gonna fail because the ionization is an instrumental component. So how we ionize our sample goes on the instrument side. I'm not sure whether or not will address that during the virtual tour, but that is something that Wonders tries to standardize to ensure that the data isn't hyper variable. Is at least somewhat controlled, which is hard to do. I want to stress, it's hard to do with FTICR. So even with all our strides, it's not 100% successful, just decently successful.
And that result field is determined based off your ionization selection.
Yes, it's based on it. Yeah, so you can't change the result field. This will change based upon your ionization and what you have as your adduct. So for example, don't do what I'm about to do. If I change the adduct to a hydrogen, you can now see it's M plus H minus P. So if that adduct is not blank, you will only have, you'll have whatever the adduct is. If it's chloride or whatever, it will change. Oh, there might be a cat in view.
Seems like we're getting more successes now, as far as I can tell in Discord.
Cool. Sweet. So again, whenever you download something from Wonders, let's see if I can actually find a smaller data set that I can download. We do have instructions that come with each Wonders. That's fine. Yeah, the fact that your database isn't named exactly the same is perfectly fine. They are identical. And sorry, but my cat is demanding attention. That's life. Be the smallest one. H.J. Andrews would be pretty small. So fortunately, I can actually show you what it looks like when you download something from ESS Dive. So when you download it, oh my God, really? When you download something from ESS Dive, simply just unzip it, and you will have a folder that looks like this. You can open up FTICR, and you'll see all the XML files that you can use. But more importantly, under the README, you will see FTICR instructions that tell you where to get all the files that you need to download, what you need, exactly what all the settings need to be, as well as providing you with an image of what the screen should look like. So if you ever forget anything or anything seems a little bit confusing, there is, anytime you download something from Wonders, you can get all of this information. And then there is some other information that you can get in here as well. So cool. Unfortunately, I am going to move on. So even though we aren't, not everyone is at the current same stage. We do need to, I want to get at least to how we actually use this data for something.
Bob, you might mention what they'll be doing in the hands-on, or at least the students will be doing in the hands-on piece this afternoon.
Yeah, so the 28 direct participants will be having a chance to actually align data later. They'll actually, I think, from my understanding, they're actually going to be downloading stuff from ESS Dive and actually aligning it themselves. They will then be using a subset of that data to analyze on KBase. So if you don't fully grasp it right now, you will have a chance later in the day to start aligning the data, and we can work on those specific errors there, here and there. So as I said, unfortunately, we do need to move on because we basically have a little under an hour, and I do want to make sure that we do cover some of the other details. So when you do get a report, whether or not you have it, fortunately, if folks do not have a report, if they didn't get a report from Formularity, under the EMSL Summer School 2020 in the repository, I did already generate the report for you. So you are able to participate in everything. You just can't, so if you didn't have any success with the original report, we got a report here. It's a variation of the report. There are some changes to it. But you will still be able to use this as a means to participate in the rest of the tutorial. Sorry, my hand's back. So if you were to even open up that report currently, the one that is on GitHub, so even if you did not get a report from Formularity, you can still look at this report along with me. You'll see that there are a couple columns that contain useful information. So the first column contains the mass, or the M/Z values. The next several columns contain information regarding stoichiometry. So things like number of carbons, hydrogens, oxygens, nitrogens, et cetera, as well as number of identified isotopic peaks. Beyond those stoichiometric columns, there is one that constitutes the composition. So this will tell you whether or not the compound is CHO, CHON, so whether or not it is a carbon, hydrogen, oxygen, nitrogen, carbon, hydrogen, oxygen. It just won't tell you the number. It won't tell you the actual formula, just what elements are present. Next is a class column. So this is similar to that discussion that was earlier today about whether or not we can identify amino sugars, things like that. This is assigning classes based upon one of those provided boundary sets. But we will actually be assigning a couple other classes as well. Next, we have a neutral mass, which I'm not going to go into. This scares me. This has to do with when it's not charged. From there, we have an error column. This is the error associated with the assigned formula. Yes, so I thought I had that mentioned. Yeah, so there is, we'll get there, we'll get there. So we have an error column. This will mention the error associated with the assigned formula and how much it deviates from the identified experimental formula. Given that we had it set to 0.5, this error should always be below 0.5 ppm. From there, we have a candidate column. This should list the number of different potential formulas that could match this mass. As you increase mass, this number increases exponentially, and there are ways to decide what is the representative formula. Not necessarily the best ways, but there are ways, and Formulary does that for you. And then from there, after that first column, after that last column, we have a whole bunch of samples that contain information about intensities, and that's about it. Again, I want to stress, even though it might be tempting to use those intensities, highly recommend always changing them to presence absence for our purposes. There are certainly experimental reasons where you might want to use those intensities, but this is not one of them. So cool. So next we're going to be moving on to actually walking through some common analysis pipelines, particularly the analysis pipeline that we use as part of Wonders. And there was, so no, they will not all be 0. They will have numbers associated with them. If they are all 0, that probably means that the database wasn't loaded correctly, or CIA wasn't checked. Or, as James says, you might have to scroll down. Where's my brain? Yes, so this data is good, and you could use this report if you wanted to, but there are some things that should be removed. And in order to do that, we use I wrote a script called FTMS R analysis, which is based off of an R package developed by folks at EMSL, awesome folks at EMSL. And in order to download, so at this point, we will need to set up a usable R environment for folks to use the scripts that I have written, again, based off other packages, of course. In the FTICR tutorial home repository, there is a file, so you should have downloaded this file as well, called install packages commands.txt. If you open it, it will have all of the packages you need to run any of the scripts that I have. You should, I've tested it, but again, things break in unexpected ways. You should be able to copy and paste this directly into R or RStudio and be able to start analyzing this, these R scripts and stuff. Should be allowing you to participate directly within these R scripts. Regarding some questions, so yes, as James says, there are going to be a lot of zeros in this data set. And that's because a lot of masses have no associated molecular formula with them. And there are a lot of reasons for that. That could be that there's database limitations. We certainly don't know all of the different combinations of organic matter that exist. It could be that they do contain other elements that we don't know. It could be that they do contain the elements that we know, but they didn't fit our user-defined filter because of, again, when you go out beyond that very basic filter, and we actually have a very liberal filter in that regard, when you go beyond that filter, you might start getting incorrect formula assignments. So like the false positive rate goes up significantly. And so there are a lot of reasons why a lot of molecular formula aren't assigned. And that's just one of the, I don't want to say, downsides, but one of the limitations of FTICRMS. But that being said, even though you can't assign formulas to all of the masses, you still get 3,000 molecular formulas in a sample. That's still pretty robust, and more sometimes. So regarding the numbers in the sample columns, those are just the peak intensities. Those are the intensities associated with the masses. And so how intense in the spectra they were. And as for our data, it is prudent to, in R, set those to 1, basically. If they have a number, or set them to 0 if it's 0, presence, absence. So is anyone having any trouble with this install packages command? I did refine it from previous installations of this. Interesting. Cannot remove prior installation of ellipsis. I've certainly not run into that error before. Waiting to hear feedback from others before I. Oh, that's also weird. So these. Yes, install from sources, that's fine. That is completely acceptable. Failed to install. Interesting. So again, there is problems. There might be variations in what R scripts folks are running. Sorry, not R scripts. What versions of R folks are running in RStudio and things like that can also impact it. Cannot remove prior installation. One thing that might be causing that is either you don't have administrative privileges, don't know why that would be, or you might have to close R to ensure that all your packages are unloaded and then restart R to ensure, yeah, as Katie is mentioning. That's my guess, is that it's probably loaded and that might be causing errors. There's no package called process X. That's weird. I don't know. I've certainly, that sounds like a dependency that I've certainly never installed myself. I'm not sure. Again, that might be an R version problem. Maybe there's newer R versions that already have that package included, something like that. And my cat's leaving. Okay, I can move. Yeah. I'm kind of pausing. We do have time, fortunately. These steps don't take very long. Once the installation is done, this is the most time-consuming step. Everything else should be really quick.
Do you want people to indicate whether they had success or not in Discord, just so you can kind of get a pulse?
Yes, I just need to know whether or not people have success, even if it's a simple, yep. Cool, awesome. I feel better, because I was very worried. Astounding silence is never a good, it could be either a good or bad sign. Sweet. So, hopefully, again, I know there might be some errors lingering, but moving beyond, in order to run the script, the first script that we're going to be working on is if you just simply double-click FTMS R analysis and open it up, you should get something that looks similar to this, where you have the required packages. You can change the sample name. You might want to change, well, actually, no, I changed that for you. So it should say K-base tutorial. Summary is equal to false, but it explains what that is. Okay, cool, that's good. This explains what it does. If you want to run that, you can, but for now you don't need that. Beyond there, if you press source from here on the FTMSR analysis, regardless of your operating system, I definitely updated that. It's fine. It's the same script. You can change the sample name to tutorial only if that works. This is just what's going to be put on the output files. So you can change that to whatever you want. So if you click source or do, what is it, Command Shift Run, Command Shift R, Control Shift Enter, things like that, depending on your operating system, you should see a window that pops up. It might appear behind a window or something, so kind of look for it if it seems like nothing's happening. But that pop-up should be asking, that pop-up should be asking where you have the report. So in this case, the report that I'm referring to is that kbase_report.csv. That is the report. Okay, Tim sounds right. I think you might be using an older FTMSR analysis script, James. Yeah, because there are two FTMS, there's actually a couple. There's one in the home page, and then there's one under the EMSL Summer School. Everything we're doing is under the EMSL Summer School except for the install command. And that's not the best organization on my part, but that's what happened. But yeah, so when you see that pop up, just select the location of the kbase_report.csv. This script will then just run, and it should run fairly quickly. What it will do is it will go through that report, kind of partition the data out, as well as remove any of those peaks, any of those masses that were associated with an isotopic signature, and any mass that exists below 200 m/z or above 900 m/z. The reason we remove isotopic signatures is that currently, under the current CIA formulation, isotopic signatures, isotopic compounds can pose some problems with the molecular formula assignment process, as well as analyzing downstream analyses. And so it's just a good idea to just remove them if they have an isotopic signature, to eliminate and control for any kinds of issues they might cause. The filter, it's fairly arbitrary. The 200 to 900 range is usually just the most, I don't want to say accurate because that's not a fair statement for the FTI CRMS, because it does go down to around 99, 97 m/z. But it's the most consistent, is what I'll say. For at least Wonders data, it behaves very consistently behind there. We can get differences and similarities using this mass range. And so if folks have had a chance to run this, kind of give a shout out, just so I can understand whether or not things are moving along or if they're completely stalled. So again, simply open it up, click source, or however you want to run your script in R if you have a different preference. And it should come up with a window. Select where that window is, in that window, select where the report is found, and you should get two files output. And I will kind of explain what those files are in a moment. Because we have about a half hour, which should be plenty of time. Because usually this half of the tutorial was the entire tutorial. And I usually had about 45 minutes to an hour. So, 45 minutes to an hour. So. Okay, that might be because you have multiple. It's probably because you're using your self-generated report, which is fair. It's fine, but the results might be a little wonky. So this is an awesome R package that I rely on super heavily, like a lot. All of my scripts, everything that I write is based off of the files generated by this R package. Now again, they're fairly modular, so I can adapt them for new reasons. But this is a package developed by Lisa Bramer et al. Over in EMSL. And it is basically a package that helps us put FTICR data into a, I don't want to say standard, but a consistent format. It keeps things consistent, regular, and easy to manage. And importantly, the way it does this is by partitioning that report into two separate files. A data file, which contains your mass by site information. So you can think of this data file, if anyone's ever worked with an OTU table before, this data file is kind of like your OTU table. It contains all of the different peaks that were identified. And remember, these are not molecular formula, these are masses. And then the different sites or time points or whatever samples that you have. The mol file contains all of the molecular information for those masses. So if molecular formula were able to be assigned, this mol file contains that information. In addition, FTMSR analysis does a lot of heavy lifting for us as well. It calculates things like NOSC. So in VGC's talk, you heard her mention NOSC, as well as Gibbs free energy, that delta G Cox. Calculates that as well. It calculates aromaticity index, which is a measure of how aromatic compounds potentially are, double bond equivalence, so how much unsaturation or saturation there is, variations of those metrics. It also assigns boundary sets. So not only does it assign that standard boundary set that we've discussed before, which are things like amino sugars, proteins, lignin, lipids, things like that, but it has two other boundary sets, one which is a modification of that first boundary set, and an entirely brand new boundary set which considers not only HC and O to C ratios, but also things like N to C and N to P ratios to get a more complete picture of the molecular landscape. But these two files, for all the scripts that I write, and if you kind of hunker through my GitHub, anything that I have published on my GitHub uses these data and mol files. And they're useful in downstream analyses, such as identifying what types of carbon are present. How do these types of carbon change through space or time? Are profiles of these carbon different and distinct to each other? So we're just going to walk through two analyses, and there are so many more analyses that can be done, but just two. We're going to be generating a couple of Van Crevelen plots. So these are these plots that plot the O to C ratios against the H to C ratios to kind of get an understanding of the molecular landscape, I like to say. And then we're also going to compare molecular properties. So things like aromaticity or just mass across all of our samples, and just get an understanding what's there. These are not in-depth analyses. These are just kind of seeds showing you how to work with this data. And there's a typo. Fixed. This is just kind of hoping to demonstrate how you can actually start working with this data and start answering questions that might be very interesting to you, rather than guiding you down a particular analysis that we think you should do or something like that. So with those data and mol files in hand, if you ran FTMS analysis.r, you can now move on to the generate_vk_plot.r. From there, all you have to do, open it up, click source, or however you want to run your script. Another window will pop up, specify the location where you have your data and mol files, and you should see two different Van Crevelen plots generated in your data set. In fact, I will do it along with you to show. So here's the thing here. Click source. See a window pop up. So don't worry about that. Another project. Where did I put this? There we go. There we go. Okay. Oh, actually. Look at me cheating. I didn't run this yet. Cool. And that runs. All right. So it finished running, and in your folder, you should now see two files. One that's titled all data, and then in this case, since I picked sample number 1, you'll see something that should be titled, unless you change the sample number, something that's SW East Fork SW43. So just to show you, this is what our Van Creveland diagram would look like for all of our data. This is what our Van Creveland diagram would look like for a single sample. And the colors indicate the different boundary sets that we've assigned based upon one of the 3 boundary sets. And you can see there's differences in between, you know, when we look at all of the data versus only one sample. Some of these differences arise. And to kind of give you some more detail, a Van Creveland diagram, so looking, this is actually an all data collected from a different plot, but they're really useful, as I said, for understanding how one sample might change from another sample in terms of its overall bulk molecular composition. So if you see a shift in the molecular formula from a very high O to C ratio in one sample to a very low O to C ratio in another sample, that might be something important worth noting. Ostensibly, a Van Creveland diagram is incredibly simple. All it is it's plotting H to C, the atomic ratio of hydrogens to carbon, against the atomic ratio of oxygens to carbon. And then a lot of folks have empirically, with some success, identified boundaries which correspond to similar, rather, I should say that correspond to overall groups of compounds. Now I want to mention, these aren't hard and fast, because again, we're only dealing with molecular formula. We have no structure, we have nothing like that. More importantly, this isn't considering things like nitrogens, sulfurs, you know, phosphoruses. And so we often use the term, and it's the most accurate we can be, is that anything within this green region here is simply a lignin-like sample. And that's about all we can actually really say. We can't say that it is a lignin and it goes this kind of lignin. We can't go that deep. We don't have that capability with this current data set. There might be ways to really dig down, but that's not what we're covering today. As I said, there are other boundary sets, and those different boundary sets and different coloring schemes might help you reveal different patterns. So did people have some success in generating a VK plot? Yes, no, maybe so. Sweet. Awesome. As I'm making lots of noise. Cool. Because what we can do now is I built into the script a way to kind of like tweak the VK plot. So not only can you tweak the threshold of, so how often you have to see some data for it to actually be included in a Van Creveland diagram, but I've also, which might, I wouldn't tweak that one. Leave this at 0. This was designed for a different data set. I don't know how this is going to behave with this data set. But more importantly, the sample number you can change. So particularly with this East Fork data, if you change that sample number to something like 4, you will get a different plot generated. And I can actually show you this. I'll change it myself to 4 and rerun it. Oh. So if you do get an error after rerunning it, sometimes you do have to clear your environment. It's a weird bug that I don't quite understand. It has to do with how the list files loads in data. Don't know why. But yeah, so you can see I created one from East Fork, and now there's a new one after setting this to 4 from Erpe, which is a river. So East Fork is a creek in Tennessee, and Erpe is a river in Berlin, Germany. And you can see there's some differences, but you can see that between these two samples in East Fork and then in Erpe, there are some variations in molecular landscape. I know what that error is. That sounds like you probably didn't get the, there might have been an error in Annette, sorry to speak specifically. It sounds like that might have been an error in loading in or installing the FTMS R analysis package. Regarding what about cross-site VK comparisons, we do have the capability to do that. Yeah, unfortunately, it does require a little bit of R scripting, but you can easily have one site colored one color and another site colored a different color. And that might give you a very direct shift in terms of molecular landscape. And it might be important to see something like that. So certainly that is very viable. And importantly, one thing I didn't mention, this entire R package underlies a web app called Frida. Oh, yes. So there are going to be some differences there. So be sure that you're always running the FTMS analysis.R script that's in the EMSL summer school. That's important. So comparing the cross-site PK plots is something that this Frida web app has direct capability in doing. And this does everything that the R script that I wrote does, because this is based off of, this is designed by the people who made the R package and uses the R package. It's a shiny app. And so it's very easy to use. I'm not going to walk through it at this moment. But you can upload the data and mol files that we've generated, or you can upload the reports yourself to this file, and then it will walk you through how to do everything up to the point where you can visualize your own VK diagrams, your own, there are many other plots that are useful with FTIC-RMS analysis, and it will walk you through each of that. So I do highly recommend folks to investigate that on their own time, as it is a very useful resource. So the next step that is pretty logical, because I do want to at least get to some of the other analyses that we can do, is looking at molecular properties. And so like my other scripts, I try to make these as user-friendly as possible, with varying degrees of success, of course. If you simply open up generate mol prop plot, and then press source, like before, a window should pop up asking you where do you have your data mol files. I do recommend clearing your environment, and if people aren't entirely sure how to do that, if you open up RStudio, there's a nice little broom. Just click that, and it should go away. So if you open up the mol prop plot, which I currently have open here, and then just run it, a window will pop up. I'm already in the window that we need. It'll generate a couple plots and give you an error. Apparently, melt changed since I wrote this script originally. It's not a problem right now. I'll update this on the package. We'll fix that. But right now, it should still work. So if we go to the folder, we now have 2 new plots, a mass distribution and molecular characteristics plot. So mass distribution looks something like that. Kind of busy. Oh, okay. Yeah, so I will fix that on my own time, but something got updated somewhere. Didn't know. But, because I did actually just rebuild R recently. But, so first, this is a very busy plot, but this is just a quick glimpse at what you can have here. This is not what I would consider publication quality. But what this is giving you a viewpoint is of the overall distribution of masses that are all in all of your samples. So this is not averaging, this is not looking at some kind of comprehensive metric. This is looking at all of the masses of all the molecular formula found in each of your samples. We have another graph that's generated, however, that actually does look at averages. So this is a bar plot that looks at the average amount of whatever the variable is. Yeah, it's not. It's a bad idea sometimes to rebuild R. This will give you an average value for each of your samples. So what I mean by that is when we look at NOSC, this isn't giving a distribution of NOSC values in our sample, but rather the average NOSC value for a sample. So we can see, for example, that this first sample, this Altamaha, 0 whatever, has on average a lower NOSC value than this. So no, this is just if out of all of the peaks that are present, sorry, out of all the molecular formula, in this case we actually do have, and all of the molecular formula that are present, that's weird. I don't know that error. Sorry, they are. Brain skipped a beat when I saw that error. Out of all the molecular peaks that are present, we're just averaging the NOSC values for molecular formula that are present. We're averaging all the NOSC values for all of those together. So if there's several hundred, several thousand molecular formula present, and we just add up the NOSC values for each of those molecular formula, divide them by the total number of molecular formula to get the average. There is utility in knowing the average NOSC value for samples, but there are also some considerations that do need to be made when doing something like this. This doesn't, I don't want to word this, this isn't, that's why I generate both of these figures, is basically what I'm trying to say, is that we have, it is prudent to understand how the distribution of these values change across the samples, but sometimes for sake of correlations, sometimes the median is necessary or the mean is necessary to get a comprehensive understanding of these systems as they relate to each other. And I want to stress, as we can see from something like these Jordan peaks here, these are going to have this NOSC value could be so low because there are so few molecular formula in the Jordan. So that's something that you do have to be cognizant of. But again, that's an experimental design question in my mind. This is more just showing kind of potential avenues for analyzing data rather than correct way. And the reason that I bring this up is that this property here, if you were to change this property to something like NOSC and rerun this script, it might give me an error. Nope, didn't give me an error. We now have a NOSC distribution plot. And you can see that even though the average is very different across the samples, the distributions actually aren't that variable in terms of NOSC. Now, what that means, that's really up to the researcher to come to some kind of conclusion for. Because it could mean anything. But it is important to still realize that there is a difference between taking an average and representing that, using that as a representative, versus understanding that there are still thousands of molecular formula in each of these samples, and they should also be considered independent, unique, things like that. Because again, we can change this to something like aromaticity. Yep. Seems to be 2 is the limit. And then so I just generated another one for aromaticity. And you can see again, distribution-wise, there's variability, but average really would exacerbate that in some ways. And again, median might be more important than average anyway. It's all about research questions. It's all about research questions. So yeah, that is actually, I think, the last hands-on component that was necessary. I do want to go for some insight. So again, the script will generate those 2 plots depending on what you selected. Here, the default, yes, quotations are important. Here, the default is mass, but you can change that mass to anything you want. That is an acceptable parameter. I list the acceptable parameters there. But just give some insight in case some folks don't remember. NOSC is the nominal oxidation state of carbon. This can give you insight into some potential thermodynamic behavior of a system. So what carbon might be more preferentially consumed or less preferentially consumed. I personally like to use NOSC a little bit because it doesn't have any standard conditions associated with it. So something like the Gibbs free energy, it's not wrong to use by any stretch of the word, but it does assume certain standard conditions. NOSC doesn't carry that assumption. And so if you're looking at a more anaerobic system, NOSC might be more appropriate. Might be. Don't want to stress. Experimental questions, man. As long as you have your question designed correctly, you're cool. Next, we have aromaticity index and modified aromaticity index, as well as double bond equivalence, as well as its many variations. These simply just reveal patterns in molecular formula properties with, while generating some inference into the structure. Now again, I want to stress this is an inference. We don't know if we have a certain aromatic value. We don't know if it is actually aromatic. We're just inferring aromaticity based upon the number of carbons, hydrogens, and oxygens and whatnot. Next, we have elemental ratios, which these are the most straightforward. These just give you insight into how your data is changing relative to itself. So the H to Cs, O to Cs, N to Cs, things like that, give you understanding of how much nitrogen relative to carbon do you have in your system. And then finally, all these metrics I want to stress are derived from the stoichiometry. And so they are entirely dependent on your molecular formula assignment. If your molecular formula assignment isn't accurate, these metrics won't be accurate. So this is just changing those things as I did. Feel free to play with that on your own leisure. We only have about 7 minutes left, so I do want to finish. There are a lot of other ways that we can look at FTICR data. The first way is the way that I kind of am most comfortable with, because I am a microbial ecologist and I like to look at how information changes across large scales or small scales, but still look at multivariate differences. And so one of the common ways that we can look at this is using something like principal component analysis or principal coordinate analysis or an NMDS. I do have example scripts for anyone, I think under my metabolomicology GitHub page. If you want to run these, you should be able to take those and kind of run with whatever data you have. But here we're looking at a Jacquard-based NMDS from the 7 rivers that you'll be working with this afternoon. And on top of this NMDS, I've actually fitted a number of the derived metrics. And what you're able to see from this is that something like the Erpe River seems to be very coordinated with the number of peaks that we have. And if you were actually count the number of peaks in the Erpe River, it is one of the more rich in carbon rivers that we have. Whereas something like the Columbia River, in this orangey yellow color here, is more associated with a NOSC value. Something about the NOSC is correlated to the Columbia River. We can see things that are like, for example, as James mentioned, earlier, we have something like the East Fork Poplar Creek, where the pore water and surface water samples are quite different from each other, whereas the pore water is actually quite similar to Altamaha in some regards, which is in blue. You can get a lot of information, just like in microbial ecology, you can get a lot of information from these multivariate analyses in FTICR. And this is me just plotting the molecular formula by site matrix. I do want to add the caveat, as I've done like 800 times before, this data has to be presence-absence. Don't do this with intensities, because you will get potentially misleading results. And it's not accurate. It's inaccurate to use intensities in this case. We can also do the transformation analysis, as VGC mentioned in her talk, where we're able to measure the mass difference between 2 molecular formula, match that mass difference to some database of known and common biochemical transformations, and get some understanding of the potential biochemical activity within this site. We do have a script that will actually allow you to use this under the FTICR tutorial GitHub as well. More importantly, one of the neat and kind of burgeoning areas that we're trying to investigate with this is that not only do we get the mass difference between these different molecular formula, but we can also start building things called transformation networks, which gives us insight into the overall potential biochemical relationships of a lot of the peaks that we have in our data set. And as James is highlighting in the Discord chat, one of the neat things is that these transformation analyses don't require molecular formulas to be assigned. They can just be worked with. They can work with just the masses. So no molecular formula are necessary. And again, while we aren't covering any of these in detail, I do have scripts in my GitHub that will allow you to take those data and mol files, and you should just be able to run them through without any kind of additional analysis. And then but as I mentioned, these biochemical transformations are kind of neat because they actually give us insight into the different biogeochemistry in systems. So for example, what we commonly see, as VGC alluded to in her talk, is that we seem to see a differential proportion of nitrogen-based transformations in the pore water as associated with the surface water. We see that both in a research site in Oregon that I'm working on, as well as some a published data set that James Stegen worked on here in Washington. Extending these analyses to other ecosystems and other transformation types will hopefully begin to reveal some generalizable principles. And then lastly, just to touch on in super brief, something that Hyun Sung will be covering in like much better detail than I could ever possibly cover. We'll be working with, we can use this FTIC-RMS data to really apply it to biogeochemical modeling. So we can start getting a measure of, I'm using the term carbon use efficiency, but it's not the same CUE as others might be familiar with, but rather how much carbon is necessary, how much of a given carbon molecular formula is necessary for some microbial organism to generate biomass, or some organism to generate biomass. This afternoon, you will actually be calculating these using Wonders-based FTMS data yourself. So that's pretty sweet. And with that, I do want to finish on time, leaving a little bit of time for questions. And again, I do realize there's some questions bouncing around Discord. There are a lot of challenges to analyzing FTIC-RMS data, like a lot. And I don't want to give a disillusion that there aren't considerations that I glossed over. But what we've done as part of Wonders is we're really trying to limit the impact that those considerations have on the end user. We want to make sure folks can take our data, use it, and answer questions that are really exciting to them. Questions that we probably couldn't even thought of if we wanted to. We do our best with moderating levels of success to provide this data as quickly as possible. Both preliminary versions, as Amy mentioned yesterday, as well, and then well-curated versions as much as we can. All the scripts that you've played around with today are going to be hosted as long as I can on GitHub. And I really do try to expand this page kind of regularly, as long as I have as much time permitting. But there are other, as I mentioned, there are other scripts on this page, on that GitHub repository, that you can use with the data that you generate today or data you generate on your own to really dig down into things. Because FTMS data is really rich, contains a lot of information, and honestly, we're only really scratching the surface right now with how we analyze that data. And so with that, I really want to thank a lot of folks. The Wonders team, they're all awesome, super helpful. James and Emily for guidance, EMSL, everyone at EMSL for helping me understand this stuff, as I came in with a okay understanding of microbial ecology and metagenomics, and had to pick up FTICR, and so I could not have gotten here if it wasn't for a lot of the folks at EMSL. And really awesome credit to the folks who helped develop Frida and FTMS analysis to really start broadening the accessibility of FTICR data, because that's always been a challenge. And of course, all the collaborators who are sweet and sent in samples for Wonders and continue to do so, because this data set wouldn't exist if it wasn't for the awesome, community science that Wonders really helps promote. And so with that, it's the end of the time. But if anyone has questions, feel free to email me. My email may not be here. I can I'll put it in the Discord channel, or just whatever. Should contact me somehow. So yeah.
Well, thanks a lot, Bob. There are, let's see, Bram has a question. You might want to ping him in Discord on that.
Okay.
So the next thing that's coming up here, we are on time, is Will Kew is going to give us a virtual tour of EMSL. Will, are you here and ready to go? I see you here.
Yeah, sorry, I just need to switch on my peripherals.
Yeah, and just before you get going, I would encourage people, maybe we should keep the discussion going in Discord as well. I don't want to distract from Will, but there's the people that are having some technical issues. Please keep throwing us those issues. We'll try to do our best to resolve them. So, all right, thanks, Will. Please take it away.
Okay, if you can hear me okay, I will get started. Let me figure out how to share my screen. Oh, that's the wrong button. So, okay, I hope this is sharing. I assume someone will yell at me if it's not. So I was asked to give a tour about the FTICR laboratory we have here in EMSL. And normally people would come to the lab, we would show them around, you'd get 5-10 minutes, see some of the equipment we have, and then move on. But we've got slightly more time today, so I want to talk a little bit more about some of the capabilities that we've got and some of their applications. So I've got a nice picture here, me looking like Vanna White, trying to show off our flagship instrument. And in the talk, I want to talk a little bit about what FTICRMS is. Bob's done an excellent job this morning of going over some of the applications of it and how you make sense of some of the data. So fortunately, I don't need to go into too much detail on that. And then I'll talk about FTICR specifically in EMSL, what that means in terms of the capabilities we've got and the application space that we're applying it in, other omics technologies in EMSL, as well as instrumentation and applications of it. And then finally, there should be some time for questions, seeing how these things go. So FTICR, if Bob didn't get to this, stands for Fourier Transform Ion Cyclotron Resonance Mass Spectrometry. It's a little bit of a mouthful, but what you really need to know is that it's essentially the gold standard in mass spectrometry. It achieves ultra high resolution, accurate exact masses, and with that, it also comes with high sensitivity. And so with all of these things, we can get molecular information. And as Bob has stressed quite well, it's not strictly quantitative, but there are ways and means of getting some quantitative information out of these mass spectrometers, but it does require due care, careful selection of standards and calibrations, for example. And the other thing which is important to note is that it's a destructive analysis. So when we're provided with a finite volume of sample, there's only so many measurements we can make, which contrasts with some of the other omics technologies we have.
Well, do you want to throw your screen into the full display mode so everybody can get a good close-up view?
It's gone, hasn't it?
Well, I can see your screen, but it's not in display. It's not in slideshow mode. Yeah.
Yeah. Okay. And then switch monitors.
And then say, yeah.
Is that better?
That's perfect. Thank you.
Okay. And you can hear me okay?
Yeah, you sound great.
So this is the main FTICR lab in EMSL. As you can see, it's quite a crowded space. You would enter on the left of this image and have a look around, and you can see that we've got 3 FTICR magnets in here. So these large cylinders are superconducting magnets, and these are 3 of our instruments we have. Any university would be very happy to have just 1 FTICR mass spectrometer. In EMSL, we're actually fortunate that we have 5 instruments, although we are only routinely using 3 for user analysis. And what I want to talk about a little bit is some of the differences between these instruments, as well as obviously a lot of their similarities. But before we get to that, I need to go over just a little bit of basics about FTICR mass spectrometry. And so I was advised to take out some of the more technical detail, which is fair enough. And so instead, we'll keep it nice and simple. The fundamental starting point of mass spectrometry is you need to generate ions. So you need to convert your sample, whether it be a liquid or a solid or your gas, into gas phase ions. And it's these ions which have charge, and it's their charge which allows us to manipulate their transmission through a mass spectrometer and allows us to detect their signal at the end. So in the case of FTICR mass spectrometry, especially coupled to electrospray ionization, which is our usual ionization source, we start at atmospheric pressure, we generate our ions, and we drag them through the instrument through various stages of ion transmission, where we maybe select mass ranges, we accumulate packets of ions, we do more transmission. And at all of these stages, we're going to higher and higher vacuum. And then eventually we send our ions into the ICR cell, into the mass analyzer, which is in the center of the magnetic field. Now, you've heard me talk about magnets quite a bit already. Why does the magnet matter, and what is the magnet about? So again, this is a photo of our 21 Tesla FTICR mass spectrometer. This is one of the flagship instruments we have in EMSL. And Tesla is a unit of the magnetic strength, essentially. It's the magnetic flux density. And when we have a 21 Tesla magnet, this is something that's about 40,000 times stronger than the Earth's magnetic field, which is only 50 microtesla. And even something like a MRI scanner, which you may have had, or you definitely have heard of, where you put a human inside a giant magnet, those magnetic fields are 20 times weaker at about 1 to 3 Tesla. So this is an incredibly strong magnet. And to get it to this level of magnetic field, we take a superconducting material, which is something which exhibits essentially zero resistance. And we put a large amount of current through it in this wound coil of the superconducting material. And that it generates a magnetic field. But to make this superconducting material have that property, we have to cool it extensively. And so in this case, we have to cool it with at least 4,000 liters of liquid helium. And we have to actively pump that so that it's as cold as just 2 degrees above absolute zero. So this is quite an engineering feat just to get the magnet to work. Why does it matter? Essentially in FTICR, bigger magnets are better, and you get higher resolution and higher sensitivity. And with 21 Tesla, we're at the highest field that's been deployed for FTICR in the world. There's only 2 instruments. Ours is one of them. The other one is in Florida. And ours is essentially a unique instrument because the magnet, whilst there's another one at the same field, the spectrometer is something that we developed in-house. And so just a little bit about our spectrometer. It consists of a whole series of electronic components. We've got various DC and RF transformers. There's signal processing units that are required. There's also a large number of pumps. One of the reasons why we decided not to go for an in-person virtual tour where I walked around the lab is because this instrument alone has got 4 roughing pumps and 5 turbo pumps, which means it's quite a noisy room to be in addition to the cryo compressors running for the magnet. And even though this magnet is actively shielded, which means the magnetic field doesn't stray really far outside of the magnet, which is very useful because otherwise we couldn't have electronics or metal tools anywhere in the same room, the stray field does go somewhat outside. And so you can see at the bottom of the photo, there's this marked line. And this is the 20 Gauss line where you would not want to cross if you've got your wallet in your pocket, your credit cards would be wiped. If you've got a pacemaker, if you've got metal implants, there could be problems. Definitely don't take metal tools into there. We have to work with specialist titanium tools when we're anywhere near the field just to make sure nothing gets stuck to the magnet. Just very quickly, we have a development instrument, which is essentially the miniature version of the same thing. The magnet was delivered by the same company, but it's a lower field strength. And this is one where we're developing the next generation of our mass spectrometer. We have this, which is really the workhorse instrument. This is our 12 Tesla Bruker Solarix. It's a commercial instrument. Even though 12 Tesla is obviously less than 21 Tesla, it still is capable of resolving highly complex mixtures. And this is our go-to instrument for any organic matter study, for dissolved organic matter or water extractable samples especially. This consists of the 12 Tesla magnet, like we've seen before, the Solarix cart, which is the spectrometer part itself. There's also a stray magnetic field from this one, although it's much smaller. This line is 5 Gauss, not 20 Gauss. And to allow us to do high throughput studies, such as necessary for the Wonder study, we have direct infusion cart capability as well. We also have a 15 Tesla instrument, so slightly higher field magnet, which gives us slightly better sensitivity and resolution. It's got a slightly newer generation of spectrometer on the front of it, as well as a slightly more advanced analyzer cell. And all of this means that this is really useful when a user sends us slightly more complex samples, perhaps a novel type of organic matter or something that's just a little bit more tricky, or if they want targeted analysis. But what it's really powerful for is when we do MALDI imaging. So one thing that Robert mentioned as well was about ionization sources. As I said, the go-to standard for us is electrospray ionization. It's the community standard for organic matter characterization. It's really good at ionizing polar molecules. So I put a couple of examples at the bottom. Something like caffeine ionizes super well in positive mode. Something like vanillic acid ionizes super well in negative mode. And basically, it's the ability to gain or lose a proton easily. It tells you if it's going to ionize in electrospray ionization. Why I emphasize this is because there are obviously other options for ionization sources, including something like photoionization, which is really commonly used in petroleomics. There's a bias and a selectivity associated with your ionization source. And there's also atmospheric pressure chemical ionization, which has a different selectivity, but also has some caveats and some problems associated with it. Just to show you some more photos, these are our routine electrospray ionization sources. So on the front of our Bruker FTICR instruments, so the ones that we use for most of our DOM analysis, we've got this electrospray source. So this is the source itself, and then this is the mass spectrometer inlet. And essentially, I would load this syringe here with acetonitrile because I'm flushing the transfer line. And you can see that that would go into this inlet. That solvent would be pushed through this silica transfer line, which is about 50 to 100 microns in internal diameter. And then that goes out through this needle where it is nebulized and formed into a fine mist. And these aerosol particles are desolvated so that we get our gas phase ions, which are then drawn into the mass spectrometer. The reason why I'm talking about cleaning it is because this is a photo of our heated ESI source, which is on the thermo instruments. So this is the same one you would get on an Orbitrap mass spectrometer. And you can see here, the needle is offset from the inlet by about a sort of 60-degree angle, as well as about maybe 2 or 3 centimeters away. And we get this pattern of residue just beneath the inlet where our excess sample has not gone into the mass spectrometer, but has formed this essentially dirt layer on the inlet. And this is why we have to be really careful when we're doing high throughput studies to obviously run things in a randomized order, to make sure that we clean regularly and run QCs and blanks regularly, because this is a contact, it's a dirty technique, and we have to be careful not to, confound our results. When we're pressed for sample and we're provided with very little sample, or very low concentration samples, such as we might have in proteomics, we can actually perform nanoflow analysis. So in this case, we can do nano electrospray ionization. This operates with maybe 10 times less flow rate, so it couples well to nanoflow liquid chromatography. It's not that well suited to high throughput studies because the emitter tip where the sample goes through is much narrower than we get in a normal electrospray ionization source. And so if there's any tiny particles at all in our sample, they can clog this sprayer and then the sample won't spray very well, so we won't get any ions. And this is another thing that matters a lot when it comes to cleaning up your samples. Obviously, if you've done a soil organic matter characterization, soil is full of particles of all sorts of different sizes, and we need to get rid of all of the larger particles, basically anything above 0.22 microns, to make sure that our sample won't clog the sprayer. And even despite that, routinely these things do clog. So there's a lot of maintenance associated. An alternative ionization source is MALDI. So this is matrix-assisted laser desorption ionization. This takes a solid state sample and we ionize it from under vacuum. And the way that this works is that we have a sample which is affixed to some target plate, some backing plate, and then we coat this in a matrix and we fire a laser at the sample and the sample matrix combined. And what happens is molecules are desorbed. Some of these molecules are ionized. The matrix especially ionizes well and desorbs well. It's tuned to absorb the laser energy. And there can be charge transfer between matrix molecules and matrix ions and our analyte ions of interest. And then those are sent into the mass spectrometer. This is routinely used in imaging analysis because the laser obviously has a discrete spot size or beam size. And so we can actually raster across our sample and get a mass spectrum for every different region of our sample, which means that not only do we get chemical information about what molecules are present, but we can decide or determine where those molecules are present in our samples. One thing that's kind of interesting in the context of this talk is that it's also possible to do direct analysis of organic matter from the solid phase. What does the moldy source look like in action? This is me loading in a target plate. So this is a rectangular stainless steel plate. It's hollowed out in the middle to allow us to load 2 standard histology slides, which are about 75 millimeters by 25 millimeters. And you can see that they're white where the matrix has been sprayed, and they're these small regions which were perhaps plant tissues or biological samples. These have been thin sectioned to about 50 microns, and then we can analyze this and get some really interesting mass spectrometry performed. But perhaps what's also interesting is we can do laser desorption ionization, so without the addition of a matrix, on organic matter. So on the left is some work that I was involved in a few years ago where we compared ESI with laser desorption ionization with matrix-assisted laser desorption ionization back in on SURFA, which is the International Humic Substances Society DOM standard, essentially. And we found that both give us really nice, really complex spectra, but they give us completely different chemical coverage. Unfortunately, Bob's already explained Van Krevelen diagrams, so I don't need to go into too much detail about that. But you can see, if you imagine your boundary sets overlaid, you have completely different chemistries revealed by these 2 different techniques. And that was on the exact same sample, no differences in handling or treatment, just whether it was solid phase with a laser or liquid phase with electrospray. On the right is a Van Krevelen diagram showing the difference between the electrospray ionization of a soil extract versus the laser desorption ionization of the whole soil. And what's really neat then is that we could study a soil without having to worry about the extraction processes, the cleanup processes, filtering out large particles, any of that. Unfortunately, this is not routine yet. It's difficult to perform well across soil, which is intrinsically heterogeneous. And so it's something that we're still trying to figure out and do method development, but there's a lot of things that are not understood, which means that it's difficult to interpret or have too much confidence in. So with all of that, what does FTICR actually tell us? Well, we can measure thousands of masses accurately simultaneously. Because it's got such a high resolution, which means the peaks are very narrow, it means we can distinguish ions with very similar masses. And therefore we can study highly complex systems, which lesser instruments might not be able to. So again, in the context of the organic matter characterization, this is our standard workflow, very general terms for soil organic matter. This is applicable to really any type of organic matter, whether that be dissolved organic matter or lignant studies, petroleomic studies, where they look at crude oil, for example, or even what I studied during my PhD, which was Scotch whiskey, which is also a complex mixture. There's these sort of 5 key stages. The first thing is the sample prep. I don't want to go too much into this, but obviously soil is heterogeneous, so we need to homogenize it somehow. We need to extract it to get it into the solution state if we want to use electrospray, and we need to clean it up. Soils routinely are full of minerals and salts, and these interfere really negatively with our ionization processes. Then when it comes to ionization, we have to select what polarity we want to go, whether it's positive and negative mode, and the specific ionization source that we want to use. The mass analysis, the instrument has a range of tuning parameters, and it's not to say that any one is correct. Often it's better to tune the instrument in the context of the masses that you're interested in, or if there's a targeted or untargeted question associated. And there's a number of acquisition parameters as well. We then have to perform data processing. There's some signal processing, which is usually automated and not thought about too much, including the Fourier transform process, data calibration, which Bob mentioned, and formula assignment. And then finally, there's data analysis. And again, this is something that you've had some experience of this morning as well. And you have to do all of these things cohesively and logically in the context of the wider question to be able to actually draw rigorous scientific conclusions. And this is another thing that Bob emphasized is with the Wonders study, is trying to make sure that all of these steps are as consistent as possible so that you can actually draw rigorous conclusions. Just a little note about formula assignment. Don't want to go too into detail. The reason why we can do it is because every isotope, every element has isotopes, and every isotope has different exact masses. So carbon-12 is defined as exactly 12, but everything else has a non-integer mass, and it has a mass defect. And this is the difference between an integer mass and the actual mass which is known. And so we can do essentially brute force combinatorial calculations where we measure a mass of something that's maybe 300, and we can determine exactly what numbers of isotopes and which isotopes would give us that mass. Unfortunately, there's error with everything and uncertainty, and so we have to narrow down our search space by using some heuristics, some chemical knowledge, and some sample knowledge. And this is why when we were talking about formularity before, you have to specify things like it's a deprotonated ion type, and these are our elemental ratios to stick within, and these are our formula tolerances to stick within. And all of those things are designed to take us from what is a very complex problem to something that's slightly more defined. But of course, note that many of those definitions are empirical or conjecture. And so there are always uncertainties associated with formula assignment. And it's very much not a solved problem. Just to highlight, there are a number of different tools. Obviously, formularity is the routine thing we use in EMSL. There's other tools, Petrolog, which is a commercial tool from Florida, ICBM Ocean, which is a DOM pipeline from Germany, MFAssign-R, which is from Lynn Mazzoleni's group, where they specialize in looking at aerosols, Ultramass Explorer, which is also from Germany, and Core MS, which is our under development tool here in EMSL, which is going to be a broad mass spectrometry processing pipeline. Look out for it soon, hopefully. 21 Tesla. You know, we have this big magnet. What can we do with it that really needs it? So here's a couple of examples from the first paper that came out in 2016. On the left is the isotopic fine structure for a peptide substance P. And the thing I want to stress about this figure on the left is that all of those peaks correspond to the same compound, the same peptide. It's just that each of those peaks is a different ion, and those ions are, whilst the same compound, different isotopic substitutions. And so we can see at natural abundance a different peak for something that has got 2 carbon-13 substituted for 2 carbon-12s, or an oxygen 18 for an oxygen 16, or 2 nitrogen 15s, for example. And what's also really impressive is in black is the observed experimental data, and in red is the simulated pattern. So not only do we observe the peaks at the right position, but at the right relative intensities. And with this isotopic fine structure analysis, we're really able to confirm essentially beyond doubt that that is the correct formula assignment for this compound. What we're perhaps more used to looking at are these broadband mass spectra of complex mixtures. This is Swanee River fulvic acid again, electrospray. And this is a fairly achievable experiment on the 21 Tesla to get well over 1 million resolving power at M over Z 3 to 400 or so. And with this, we can resolve peaks that are separated by just 0.4 millidaltons, which is less than the mass of an electron, which is about 0.5 millidaltons. So this is really incredibly high resolution that you couldn't dream to achieve on something like a QTOF type mass spectrometer. One thing that is my area of interest perhaps is we have our mass spectrum that looks like this. It covers a large range of masses. This is something that's been known for the best part of 20 years. When we look in at slightly closer, say between 600 and 614, we see this repeating series of peaks clustered at even and odd masses. Where it's an odd mass and it's more intense peaks, these are our carbon-12 monoisotopic peaks, and the next one across is our carbon-13 isotopolog peaks. And if we zoom into a single nominal mass, so 607, we can see a series of peaks. And so at every nominal mass, there are many peaks, and therefore we get thousands of peaks across the entire spectrum. We have these repeating distances between peaks. In this case, this mass difference of 36 millidaltons corresponds to the substitution of an oxygen, which nominally weighs 16, and a CH4, which also nominally weighs 16. And you can see, therefore, that the majority of these species are going to be related in molecular formula, except for this substitution. Again, to stress what Bob mentioned, formula is not structure. And to really highlight one ion could be many isomers. So that peak here may be many different things which share the same molecular formula but have completely different structures. And again, the intensity of these peaks is not quantitative with this measurement the way that we routinely do it. But one more thing is, between any two of those peaks, those big intense peaks we could assign, there were all of these minor features which you would just write off as noise. But when we do more detailed experiments, we can actually see that no, those are real features. And so even between those two peaks, which are only 30 millidaltons apart, there's at least another half a dozen or so ions which we can detect. And these correspond to things containing heteroatoms like sulfur or nitrogen, or containing additional isotopic log substitutions. And so even when we use 21 Tesla, and even when we do really careful mass spectrometry, there are still features in these really complex samples which might not be detected and might not be resolved. So it's really, it's, you know, complex problem with so many unknowns even to this day. Very quickly as well, everything we discussed about mass spectrometry is based on essentially intact molecules and molecular ions, which gives us a mass which we can turn to a molecular formula. It's important to note that mass spectrometry also has a long history of being used for fragmentation, where if we can break up a molecule and measure the masses of all of its chunks, we can then infer more structural features. For example, if one of those chunk masses corresponds to the mass of a sugar unit, we can assume that there was a sugar, for example, in our structure. And this allows us to get more information about structural features. And in FTICR, we can actually do it in a number of different parts of the mass spectrometer and with a number of different technologies. A little bit more about the data analysis. So in EMSL, we have two software development routines for formula assignment. The current public one is Formularity. It does the assignment, it does recalibration and alignment, as you've seen already. And then Core MS, which is our future tool. It's under development. And the aim for Core MS is to be a universal analysis software, which will do all of the processing steps, allowing you to go from the raw instrumental data all the way through to molecular formula with confidence associated with your molecular formulas. We also have FRIDA and FTMS-R analysis, which I don't need to talk about too much because Bob's already mentioned some of those. And then moving on from FTICR, I was also asked to briefly talk about some of the other omics that we can perform here in EMSL. So the other instrument that I use routinely is nuclear magnetic resonance, or NMR. In a very superficial sense, it's kind of like FTICR in that it relies on superconducting magnets, except this time they're vertical as opposed to horizontal. So you can see in this photo, we've got a large magnet in the background. This is a 17 Tesla magnet, so it's still an incredibly strong magnet. And this is a non-contact, non-destructive technique. And so our sample, which may only be half a milliliter or so, goes into this glass tube, and we then can probe the sample this way. This is quantitative and it provides structural information for a specific nucleus, a specific isotope, such as hydrogen-1 or carbon-13. There are caveats. The data tends to be much more complicated than mass spectrometry. The sensitivity and the resolution are definitely limiting factors, especially compared to mass spectrometry. On the advantages side, though, there's fewer biases than in mass spec. It gives a much more reproducible result. It's possible to do de novo structural elucidation. We don't need the databases or standards to be able to solve a structure with NMR, although we almost always will still need the mass or the molecular formula to determine what components are in there. And then what's really neat in this context of complex mixtures is we can study intact raw samples. So we can take a DOM water sample, put it straight in an NMR tube and analyze it without having to worry about going through as many cleanup steps, for example. This is a very late addition to this slide, but basically one thing that was mentioned was how do you use all of these techniques in the same sort of study? So this is a really neat paper from 2015 where they compared and combined NMR spectroscopy with FTICR mass spectrometry to study the organic matter they managed to extract from one of the Murchison meteorites. And you can see the NMR spectra here. You don't need to worry too much about it. Suffice it to say that we've got a series of sharp features and these unresolved humps. When we do traditional metabolomics, we tend to see more sharp features which are easier to identify. And when we do things like DOM, especially SURFA, there's almost no sharp features and it's just unresolved humps. So the data analysis between metabolomics NMR and organic matter NMR are pretty distinct and pretty different workflows, but we can still get really interesting quantitative structural information either way. We also use gas chromatography. This is perhaps something you're much more familiar with. This allows us to separate molecules prior to the mass spectrometry analysis. It improves the chemical coverage. It's almost always performed with fragmentation, so we get structural information, but it's only suitable when we have volatile compounds or compounds that can be made volatile through derivatization. So there's always questions about how representative the sample is by the time it gets to the mass spectrometer versus how it was in the sample. And almost all of GCMS is done with databases and standards. So if you don't have the compound already analyzed in a database, you probably won't identify the compound. That said, it can be used quantitatively with calibration curves, and it can give very good sensitivity. What's also incredibly powerful and really commonly used around the world is liquid chromatography. So this, again, we separate our compounds prior to their analysis in the mass spectrometer, and this gives us much better coverage. We have the ability to perform a lot of online fragmentation, and therefore we can get a lot of structural information. It's really powerful, but development can be challenging, especially for really complex mixtures. So things like organic matter are essentially chromatographically inseparable, and we cannot take something like Swanee River fulvic acid and put it through a column and get individual compounds out. It is just too complicated, and what happens is we get a smear across time in our chromatogram of complex mixtures at every point. But with really clever chromatography, we may be able to separate out classes of compounds. It is, however, really widely used in metabolomics studies, lipidomics, and proteomics especially. So the photo here highlighting the LC system coupled to one of our Orbitrap mass spectrometers. There's some really neat developments in ion mobility, which is in a way another separation technique, except this time we're taking ions and we're separating them in the gas phase. And this separates them based on their size and their charge. It's really fast technique, and it allows us to separate isomers. However, the resolution is perhaps limited in some respects, but even so, we have this figure from a recent book chapter, and they applied TIMS, so trapped ion mobility spectroscopy, with Fourier transform ICR mass spec, and they analyzed Swanee River fulvic acid, and they got this standard, normal distribution of complex mixtures. And looking at a single mass, a single molecular formula, they were able to deconvolve at least 9 different isomeric structures for that one compound. So just to show just how complex these complex mixtures really are. Typically, however, ion mobility is coupled to fast mass specs like QTOF, and often with LC coupled to the front as well. So you end up with this multi-dimensional data, which gives you really powerful separation techniques. There's also an effort underway in PNNL for the standards-free metabolomics workflows. And so this is where we want to take basically in most of the mass spec metabolomics workflows, you need a database of your compounds. But all of the known databases of compounds maybe cover a few 100,000 molecules. And if we look at the number of compounds that you could have in a lab that you could get your own reference data for, you're looking at just thousands. But chemical space will cover maybe 10 to the 30 different compounds. And so we can use a combination of machine learning and quantum chemical predictions coupled with some experimental data to build up databases of as wide a chemical space as we want. And then we can use that to do our molecular identification for unknown metabolomics. And so this is a really powerful development being led by Tom Metz here in PNNL. We also have the ability to do proteomics on incredibly small sample volumes. So one of the big challenges we have in a lot of these analytical sciences is we're so limited with samples. And normally in proteomics, where we want to understand more about the proteome of our sample, we need thousands of cells or more to get a deep quantitative profiling. But with the nanopot system, which is microfluidic devices where we do on-spot processing prior to analysis, we can get a spectra for just a single cell. which allows us to work with really limited samples, but also allows us to understand differences between individual cells. And so this is a really powerful and exciting technique. And so with that, I think I've rushed through enough about mass spec and omics here in EMSL. And I put my email there at the bottom if you have any questions. And I'm also on the Discord. And if you have any questions here in the chat.
So I was muted. Thanks a lot, Will. That was really cool. So I'm seeing a couple questions. We have a few minutes here. We've been answering questions kind of along the way. There was a question about the ionization detection when performing MALDI FTIC-RMS with respect to salts and the impact of salts since they cannot be removed like they could on an aqueous extract by a solid phase extraction. Do you have thoughts on that?
Yeah, that's an excellent question. So one of the driving reasons to do MALDI directly from the soils or the soil extracts is so you don't have to remove necessarily these salts. And we know that organic matter binds to minerals and all of these components in the soil. And perhaps if we could desorb it directly from these salts or these minerals, that would give us interesting chemical information. I've got like 4 caveats after that. The first of which is we don't really understand how the laser interacts with the molecules alone, let alone how it interacts when those molecules are bound or interacting with salts. So it might be that there are other interferences happening in there. So we've definitely noticed that it's more difficult to get good spectra from homogenized soil than from a homogenized extract of soil. As for whether or not the salts interfere the same way they do in ESI, where you just get lots of salt clusters, it doesn't seem to be as such a problem because these salts don't themselves absorb the laser energy very well. They don't desorb very well. They tend to be pretty inert in that regard. So we don't get big salt clusters like we do in ESI. And in fact, when you do normal MALDI imaging, it's quite common to include in your matrix some amount of salt to help boost the efficiency of these things binding and desorbing. I think that's 4 different answers to 2 different questions, but maybe it helps.
Thank you. Let's see, there's a question in the Zoom chat here. Is the formula assignment methods you mentioned for FTICR useful for other techniques such as GC or LC?
Yeah, so it's always a continuum. And it's kind of related to a question that came up on the Discord, which is, what formula tolerance is correct to use? If you've got a simple, well-resolved spectrum and you know a lot of things about it, and there are big distances between your peaks, you can use wider tolerances, which means, conversely, you could use a lower resolution mass spectrometer. So the way I read the question is more, could I use it with a QTOF type instrument or an Orbitrap type instrument? So Orbitrap, definitely yes. A lot of people do their organic matter characterization with Orbitraps. QTOFs is a gray area because the resolving power is so much lower than FTICR or Orbitrap. It becomes much more difficult to have any strict confidence based on mass alone. And this is why when we do things like LC coupled to QTOF for metabolomics, we cross-reference the retention time in our chromatography, and we cross-reference the fragmentation pattern if possible. So therefore, we've got additional piece of information. And it's always the case that if you want to get confidence in your assignments, you need as much extra information as possible. But the principle is essentially the same for formula assignment.
Thanks, Will. Another question I saw on Discord is, what is the TOC limit for whole soils? I think this might have to do with MALDI, though I'm not 100% sure.
Yeah, so in the case of MALDI, it's an interesting question. The worst soil, sorry, worst is the wrong word. All soils are beautiful. The lowest carbon soil I worked with was 0.05% TOC. It was essentially sand. It was a very high mineral soil. And that I could get signal for, but that was the time when these salts start to dominate the spectrum simply because there's nothing else. However, the really nice data I showed was about 4% TOC, and that worked beautifully. So I would say somewhere around 0.5 to 1% is probably an acceptable low limit. And if you want to go lower than that, you might have challenges. The flip side is as well, I found with 19% TOC soil, there was too much signal, and I couldn't get few enough ions in the analyzer that I could get a high resolution spectrum. It was just too complicated a mass spectrum with that much TOC. It's kind of a weird problem to have.
Just to build on that answer, are there ways of sort of diluting a solid sample with a organic matter free porous media to avoid the oversaturation problem?
That's a really good idea. We haven't tried that. I don't see any reason why not. The challenge would be, the big challenge we have in the moldy stuff is inhomogeneity or heterogeneity, whatever word you want to use for the sample. Every area the laser scans, if it is different for whatever reason, we will get different amounts of ions generated. And the mass spectrometry, to get really high resolution results, relies on us having essentially the same number of ions in the analyzer every single scan when we co-add many, many scans. So if we go from really high signal region to really low signal region, our peak positions will shift slightly, or they will broaden, or they will split. And so if we do decide to try and dilute it, say with some, plain sand or something, we have to figure out a way to homogenize that well enough on a scale of microns.
That seems challenging.
Yeah. It's a good idea though. I like it.
Something to think about. So I'm not seeing any other, wait. Oh, they say, okay, here's a new question in Zoom. For FTIC-RMS, would you say you have the same or similar TOC level requirements, I think with respect to the concentrations?
Yeah, so when we do the electrospray on the soil samples, if the TOC is too low in the soil, I think our rough boundary condition is something like, if it's above 10% TOC, like it's a really rich soil or it's a peat soil, for example, we can get away with extracting just a few 100 milligrams of that sample and getting a liquid which is going to be about concentrated enough. If it's a lower TOC soil, we will extract a larger volume, a larger mass of the raw soil to compensate for that. So at really low TOC soils, we might have to use 2 grams of soil or more to get enough extract and enough dissolved organic matter, which then we can concentrate up by using solid phase extraction, for example, or lyophilization.
Yeah, and one thing, I said some of this in the Discord related to concentrations. For Wonders things, at least, what we do is all of our water samples, they all get normalized to 1.5 milligrams per liter of DOC prior to going through the solid phase extraction. And the same thing with our water phase extractions. When we have sediments, we extract them in water, we measure the DOC concentration of that extraction, and we normalize all of those to the same thing, to 1.5 milligrams per liter. That then goes through the solid phase extraction, and I think that's increasing the concentration up to about, I think it's 9 milligrams per liter, 9 to 10 if I remember correctly. So we've tried hard to remove variation in the results from the FTICR, at least, from initial concentration differences across samples. So that's one normalization element that we've tried to do. Any other? I'm not seeing any other questions. We're basically right on time, 1 minute early. Let's see. So I will remind myself of the agenda. So right now we have a half an hour break, and at 12:30 we'll come back. Hyun Saab will give us some kind of a science overview for substrate explicit modeling, and lead us then into a tutorial related to lambda theory. And after that, we'll do a hands-on piece. As I mentioned earlier, but I want to reiterate that at the hands-on piece, the schedule has changed. That will start at 2 o'clock instead of 2:30. So Tim, is there something you wanted to end with for the moment?
Yeah, I just wanted to also note that, so while we're giving you a little half hour here to grab some lunch or whatever it is you want to do in the time zone that you're in, just wanted to point out that people haven't really been using too much in Discord the voice channels. So there are voice channels. If you jump into a voice channel and say hi, you can have an impromptu voice chat, or you can even turn on your video and have a face-to-face video chat in those channels. So maybe during this half hour while we're not running Zoom, is a good time for you to jump into a voice channel. I know Linda's been hanging out in the press channel, and I'll go hang out in the meet and greet once I've grabbed my lunch, and would love to talk with anybody who wants to just say hi.
Very good. Great call out on that. So we'll see you all at 12:30 then, if not in a voice or a video channel. Okay, see you all.